The future of research depends on it, and patients are counting on it. We also meet at a moment of real strain. The recent government shutdown, ongoing resource constraints have deeply affected our colleagues at the FDA, NIH, and NCI, the institutions that form the backbone of biomedical research. To all of you who continue to do this work under extraordinary circumstances, we thank you. Your resilience and integrity are the reason science keeps moving forward. We are deeply appreciative. We must remain steadfast in our support for science, for evidence, and for one another, because when we protect the scientific process, we protect patients, and that's what we are doing this for. The fact that more than 300 of you are here in person and over 1,000 joining online shows how strong and united this community is. None of this progress happens alone. We need all of you to do this together. To our partners in academia, industry, and advocacy, and the patient community, thank you for your courage, your creativity, and your collaboration. And to patients and families who guide everything we do, you're the reason we are here. Let's continue to challenge the old framework, align on solutions, and move faster, because patients cannot wait. Let's go to work. It's now my great pleasure to introduce Dr. Jeff Allen, who will share how Friends is leading the efforts to modernize evidence generation and strengthen collaboration across sectors. We cannot do this without you.
Good morning. I'd like to add my thanks to Ellen for everyone for joining us this morning and throughout the day. As Ellen described, we have a great series of sessions lined up for today's meeting. All advanced registrants for today's sessions should have received copies of the white papers that have been prepared by our working groups addressing the issues for each of today's sessions. Those can also be accessed by the QR codes on the meeting material that should be found at your seat for additional copies. To put each of these white papers together, leading experts have been meeting since early this summer to begin to work through the topics of today's meetings and begin to formulate proposed solutions and considerations that will be presented as part of today's discussions. I want to certainly thank all the members of those working groups for working closely with us, in particular those from the NCI and the FDA who aren't able to join us today given the government shutdown. We want to encourage audience participation in today's meeting as well. So when it's time, please utilize the Q&A link on your agenda, which you can log into, or to utilize the chat for those of you that are participating online today. Your questions will be collected internally by our team here and then relayed to each of the session moderators so that they can be raised with the panelists to address them in real time with members of each session's working groups. I'll remind all of those participating today, either virtually or here in person, that you can add to the discussion by following us on LinkedIn and X and joining the conversation using hashtag FriendsAM25. I'm told that we are in the process of restarting the Wi-Fi signal, so you will have to log out and log back in order to be connected to the Wi-Fi in the room. So please do so now before you need to log into the QR code so that you are ready to go as the discussion begins. But to get us started today, I'd like to welcome the panelists for the first session to come on up and grab a seat on the stage here. And as they get started, I'll turn things over to Dr. Alan Melemd, Chief Medical Officer of Chimerix, a Jazz Pharmaceutical company, to provide us with an opening presentation and moderator for our first discussion session today. So thank you all for your continued help.
Thank you. Before I start, I want to thank first Ellen and Jeff for first inviting me here. I'm humbled to be in your presence and for bringing such a great group of people here. It's pretty amazing to see all the leaders around here that are here to really change policy and procedures and help ultimately get patients drugs quicker. And that is one of our panels here. I also want to thank, as Ellen had mentioned, the FDA participants who are critical in this white paper that we have, which is seamless trial designs in rare cancers. Unfortunately, they cannot be here today, and I'm not sure I can actually say their name, so I won't. But I want to thank them for participating. They've really helped change the white paper and really tried to strive to get this to patients quickly. I also want to thank Bernadette Navarro, who really helped orchestrate a bunch of cats in a room to get us all together to find one policy, which I do think is a good white paper for that. Let me go to the first slide, please. Can you put that up? I can't see it fully. I need my glasses on here. Oh, that's better. I can now see. It's not advancing. There it goes. Rare cancers have unique challenges for drug development, requiring innovative approaches to efficiently generate meaningful data to get drugs approved for patients. Challenges with rare disease are several. First of all, they're small populations. As a small population, you have much more challenge with heterogeneous issues, and they're harder to find, and they're harder to get these patients specific in clinical trials. In addition, they have much more complex designs, so there isn't a single biomarker in often these diseases, which makes development even more challenging for these patients. Thank you. Seamless trials can help integrate safety, dosing, and dose optimization to maximize effect in these patients. Our ongoing working group helped to find better ways to streamline this into more of a seamless trial design to get drugs hopefully approved to patients in a quicker fashion. Those in this room, I don't need to go through this, so I'm going to go quickly. What is a typical traditional way for a traditional design? You start with the phase 1, which can have multiple dose levels where you're looking at MTD or clinical pharmacology. And then you pause and you wait, and then you evaluate. And then you go to a phase 2 trial design, again, where you're looking at a randomized dose, maybe for dose optimization, seeing signs of efficacy, and then you wait again and evaluate, and then start a phase 3 trial. As you can see, intentionally, there's a lot of pauses in these, and it actually slows down the development. Is there a way you can actually combine these phases, specifically in the rare population, where you can get these to patients quicker? What we are presenting here is more of a seamless approach, where you can take a phase 1-2 trial and lead to that to find the dose optimization first, and that can lead to a registrational phase 3 study. Or you can start with a phase 2 trial and go right into a 2-3 design. And the last example would be where you have a single trial design, which includes phase 1, 2, and 3 for registrational intent. Now, what I'm going to tell you right here is that if you're hoping to get the roadmap for your specific drug, it's not going to happen. We give you examples here of ways to think about this and how you can actually accelerate and give you some ways to do this. But one of the keys here are there ways that you can do this. And the panel had advocates, FDA, pharmacy, biopharma, and large pharma to find ways to move this quicker. And FDA has been very encouraging us to find ways to move this quicker to patients. This includes patient advocates from the beginning, why the trials are important, why the patients are important, and getting their perspective is very important early in the trial design. Obviously, the regulatory is critical. What's the right endpoint? What's the comparator? How to move forward with this? Safety, dose optimization. As we know, more and more patients need to have dose optimization in this. How can you build this on an early trial design to find the right dose to move forward? I'm going to put my statisticians on there. You can't just have an open-ended study either. You need to have some pre-specifications, so if you do see a signal, you can actually then claim significance and help move forward from a regulatory perspective. And lastly, you need to find the right endpoints, whether it's response rate, progression-free survival, to move this forward for these patients. We have 3 examples in this white paper. One of them is with a FGFR2 inhibitor, where they used a seamless phase 1-2 design to find the optimal dose. They actually studied 7 dose levels, and then they went up to a dose optimization for to find which one it was the right dose to move forward, and that subsequently into a phase 3 study. We also had a seamless trial design where you looked at all these together, a phase 1, 2, 3, essentially a registrational trial. That's the ARROW trial, which is a RET inhibitor. It's in the white paper, and the panelists will discuss these more thoroughly. And lastly, where you have just a pivotal, you know your phase 1 dose, you do a 2-3 design where there's a dose optimization with 3 doses, you pick the best dose, and you go right into a phase 3 trial. The first 2 examples have actually resulted in FDA approval. The last one is ongoing in a phase 3 registration right now. Effective implementation of this can hopefully get patients drugs quicker and alignment through both regulatory and hopefully a reimbursement perspective. I'm going to now introduce the panel members. I got to take out my glasses for this one. Harinde Abesenge. Oh God, I missed that one, didn't I?
You're fine.
I did. Lola Fashoyan-Ajay. Harindra is Vice President, Head of Oncology, Global Regulatory Affairs, Johnson & Johnson. Lola Fashoyan-Ajay is Senior Vice President, Head of Regulatory Oncology, Cell and Gene, Parexel International. Misha Mehta, Forensic Cancer Research Advisory Advocate. Christopher Turner, Chief Medical Officer, Nuvalent. Yuan Yuan, Chair and Professor, Department of Biostats, University of Texas MD Anderson Cancer Center. Thank you, everybody. I'll let you know that there will be time at the end to ask questions. Please use the QR code or use the chat box in your, if you're online, to ask a question. And I'll just start with the first one for the panel. When we initially started this, we discussed how should we define rare diseases. And we decided that we were not going to define rare diseases. And I'd like to have a little conversation on why we decided not to put the rare into the white paper. Chris, can you start with that one?
Sure. So we had a lot of discussion about this, of what does rare mean. One of the main ways you start thinking about it is prevalence based on standard histopathology. But that doesn't always cover everything right now. We have subgroup populations that are defined by biomarkers or specific disease characteristics. And so it's really broader than just prevalence. One example that we've been involved in is lung cancer, where lung cancer itself is not rare, but you look at subpopulations that are biomarker selected, such as RET positive lung cancer or ROS1, each of these like 1 to 2% of lung cancer, then you can, rare is appropriate in those settings.
Lola, would you mind giving us your perspective?
Yeah, I mean, I would agree with what has just been said. I think historically, the rare disease or rare condition definition has stemmed from the Orphan Drug Act, which was put into law in the 1980s. And in in that statute, the definition of a rare disease or rare condition is one in which the prevalence of the disease or condition occurs in 200 or fewer individuals in the U.S. And as has just been mentioned, the way we define cancer, currently is based on, many times, on molecular aberrations. And so most cancers really fall within the scope of that definition. And so really the critical considerations for, defining a setting in which the number of patients may preclude an extensive drug development program is really thinking about things including the prevalence, but also the feasibility of doing the study and the clinical context in which the study is being, or the program is being developed, because those factors really impact how quickly and efficiently one can complete the study. And the regulators are keen to have the right study design in the right setting that answers the critical clinical, scientific, and regulatory questions, and they're also keen to have those studies be completed quickly because there are other implications for having trials that go on for a long time. And so I think part of the burden for drug manufacturers or sponsors is really to be very clear about the factors that they're taking into account in selecting a particular strategy for their development program. And that includes also other competitive, other trials that are happening in the clinical space, which could significantly limit the number of patients available.
Thank you, Lola. Misha, from a patient advocate perspective, can you give me your point of view of that?
Yeah, so from a patient advocate perspective, right, rare disease, rare, I would define it as an experience. Having lost my son to a rare pediatric brain tumor, it is truly an experience, and it depends on how you define rare, and it changes who's defining that rare. For patient advocates, it could be rare in terms of biology, like Chris talked about. It could be in terms of age, depending on what age group you fall in, those cancers could be rare. It could be based on geographic location. Something that I just learned very recently when I was talking to my son's neuro-oncologist, like there are no rare diseases in China. That is what she told me, just the sheer number of patient population. So it kind of like the definition of rare is so fluctuation. It fluctuates a lot, and we need to make sure that understand that for rare disease, and I can speak only to the pediatric brain tumor community, that the traditional standpoints of what a trial looks like, those frameworks don't work. We need to make sure that every child or every participant and every disease type or every data point is accounted for. And when you do that, then the numbers are not so rare.
Thank you. What are the characteristics in an oncology study, and why might these approaches be specifically important in a rare population? Harindra?
Yeah, good morning. Really great to be here. So as Alan mentioned in his opening, the characteristics of a seamless trial is instead of having those distinct and separate phases for your sequential development in phase 1, 2, 3, for your dose finding, dose optimization, expansion cohorts for safety and efficacy, you integrate the different phases into one cohesive framework, and it can even be a single protocol. We focus specifically on rare cancers because small and heterogeneous patient populations can limit the feasibility in conducting traditional randomized trials in separate and distinct phases. So although these challenges with small patient numbers are not unique for rare cancers, they are often magnified, and that's why we focused specifically on rare cancers for the purpose of this white paper. Also, when you conduct trials in distinct separate phases, the lead times can be long. There are usually pauses as you learn about the data from one trial and you transition to the next. And this, of course, can take time, whereas if you have a seamless design, you can conduct the trial in one continuum, reducing sort of the lead times, and you can have both operational efficiency. And ultimately, this can lead to early access for patients for novel oncology drug therapies, and of course, that's a really good thing if we can end up with having early access for patients. Of course, there are challenges with seamless design. It requires a great deal of upfront planning. So you don't have the luxury of waiting until your phase 1 or phase 2 trial concludes to be able to design the next phase. You have to, the idea is you sort of design it at the onset. And things like your dose, your patient population, sample size, endpoints have to be defined well in advance. In addition, it's also important that you preserve the integrity of your trial and statistical rigor. And so there are challenges also associated with planning all of that upfront in your seamless trial design. There are other challenges, and I think we'll get through some of that during the course of the conversation.
Chris, what are some of the challenges you see in these rare cancers?
Yeah, one of the things that you both have mentioned already is just there's no single design there, whether it's a phase 1-2, whether it's a phase 2-3, whether it's a phase 1-2-3 combined. But in these rare population, more so than any other trial or disease indication, every single patient is critical. You don't have the luxury of having a whole reservoir of patients that you can tap into for trial after trial. So you really have to make every single patient count. And so it's thinking about, if you're talking about a phase 1-2 trial, typical first-in-human phase 1 is a dose-finding trial where you're really trying to find an optimal dose and you're focusing on safety. Well, in a seamless design, you also need to be focusing on, how to use other aspects of that patient's data. And so activity can be important there. And we can talk more about that a little bit later. But you have to really, get the most out of every single patient on trial and really not use their data for a single purpose, but think in advance about all the different ways that you can use that patient's data.
Thank you. Lola, what about your perspective? You have a unique perspective, having been FDA perspective and now from another CRO perspective.
I mean, I think I would agree with all of what has been said. I don't know that there's any, I think the considerations are the same irrespective of which side you're on. I would just say that I think the seamless or adaptive design studies approach is something that has been used a lot in oncology, and I think we're seeing increasing use of that even for cancers that are not rare. I think the critical thing to add to what has already been said is really the importance of knowing a priori where the key decision points will be and what the critical adaptations to the study will be and what data would inform those changes, right? So are the adaptations going to be based on data that's emerging from the ongoing study in that particular trial, or will it also be based upon data information that's external to the trial, and what are the key decision points and what will be the data that support those decisions are important considerations.
Thank you. Chris, you had mentioned there are many different approaches, a phase 1, a 2-3. What are the factors in determining which you would pick in your development perspective?
Yeah, so it really depends on your molecule, what the characteristics are, what the patient population you're going after. So you factor all of that in, but it certainly is challenging because you need to think in advance about the different components more in the rigor. I had mentioned earlier the phase 1 patients for dose escalation are not just used for safety, but you may be using them for activity as well too. So you have to potentially, if it's a solid tumor, build in independent RECIST review of that data, which you typically would not do for a standard phase 1 trial. Or patient-reported outcomes, something you usually do in a phase 3 study or a late-stage phase 2. Maybe you want to start thinking about that in the phase 1 portion of the study. So you really have to have a holistic view about, begin with the end in mind, where you're going to be at the end, and don't just think of this as, a first-in-human study.
Ying, can you comment on the importance of pre-specifications and why it's critical in certain steps?
Yeah, so for adaptive design, it's critical to specify all the adaptive rule. And it's clear, specified, stated in FDA's guidance for adaptive design. And adaptation all depends on the interim data, which we certainly don't know when we design the trial. And so in this case, the simulation becomes a very important tool to evaluate design operating characteristic. And in general, we need to run an extensive simulation, based on a variety of scenario which covers all possible adaptation to ensure the trial have a desirable operating characteristic and ensure study integrity.
Thank you. I'd like to go back to Misha about the perspective from an advocate perspective in this decision.
Yeah, so from an advocate perspective, right, like we want to make sure that there are no rigid phases that you have to go through. For a condition that my son had, right, the prognosis is 9 to 12 months after you're diagnosed. There isn't much enough time there that you have to wait for a phase 1 study to be completed, and then the interim period where you're waiting for a phase 2 to start. We've waited for that time, and we know that we don't have the luxury of the time. So in a sense, these seamless trials make so much sense that if you do a phase 1 and a phase 2 together, where you're doing the safety as well as the dosing of it, and know if there is a positive signal there, you quickly then expand the cohort, and then you enroll these kids. I'm talking from pediatric perspective, but even for rare disease patients, right, you can quickly enroll them very rapidly into this. And I think there is so much positive and perspective that needs to happen in this case, right? And I would also like to put in another layer onto what has already been discussed, which is the platform trials. If you think about having a seamless platform trial, I think that is a combination that would really make more sense, in a sense that you don't have to create a new infrastructure to test each drug individually, where you can have testing multiple drugs in a same arm, in a multi-arm study, where you're comparing one arm against the other arm. You're using the shared infrastructure. You're using the same protocol for recruitment, as well as for eligibility criteria, as well as for sorry, I'm blanking out on the word, as well as for, it'll come to me, but I'll talk about another point. But in a sense that you want to make sure that you're saving money, you're saving resources, plus you're expediting research in the same sense. The point that I want to make is that in most of the cases, the control arms are also same. So in a rare population, that is really, really important, where you have one set of control arms and you're comparing multiple different treatments against one another, or against a common control arm, which is, I think, is a powerful treatment it's a powerful testament for a seamless trial.
Thank you. During the white paper discussion, we had a lot of discussion about when to bring in the advocates, how to balance the scientific value, the burden for patients. Misha, do you have any examples of bringing in patient advocates to regulatory discussions? And curious what your thoughts are there.
Yeah, so there have been, that I know of, there have been really two very big success cases in pediatric brain tumor space. One was the approval of tovorafenib, which is now known as Ojemda, which is for low-grade glioma. And Chris knows a lot about that drug already, but that was highly patient-led. In fact, that program started because it was a group of patients that have come together and that have funded that program, that then went and became a company called Day One Biopharmaceuticals, and that led to an approval. The other example, Alan, that you are quite familiar with is the example of Modeso, which the generic name is ONC201, which had a lot of patient input in that, not only in the drug approval phases also, but even early on in terms of having expanded access to get to the kids that needed it, as well as to identify what are the correlative studies that you could be looking for signal going beyond the 25% reduction of the tumor size on an MRI, or even just the overall progression-free survival, right? So you're actually looking at daily living, you're quantifying quality of life assessment, and those are the inputs that I believe the patient advocates gave Alan in the case of Modeso.
From a pharmaceutical perspective and a regular perspective, Harinder, can you give me a comment on how you bring this in and what your thoughts are on that?
Sure. Yeah. So first of all, it was really wonderful to have Misha part of the working group. I mean, we couldn't have done this in a thoughtful and meaningful way unless we had the patient advocacy and the patient support perspective. And I'd say even from an industry perspective, we feel that it's extremely important to have patients, families, patient advocates involved in the clinical trial development and design process, regardless actually as to whether it's a seamless trial or not. Patients can provide a unique perspective into the overall design that go beyond the data, because they fully understand the experience of having gone through the disease themselves and having experienced different therapeutic options, ones that work, ones that didn't work, ones that had really some unacceptable toxicities and side effect profiles. So because they are in a unique position, getting their input is extremely important. And I know that now many companies, including ours, we do include patients and consult with patients on our trial designs to get that perspective, which is extremely valuable, and I think we should be doing even more of that. From a regulatory decision-making point of view, I know that the open public hearing portion of an advisory committee meeting, in our case in oncology, the ODACs, the public hearing session where patients come and share their perspectives, their experiences, is an extremely valuable component, and also is factored into the voting, is factored into the FDA's decision-making process. But I also like the idea that was put forward of having patients also join meetings with FDA and other health agencies, especially with seamless trials, because there's a lot of upfront planning. You have to balance things like pre-specification with some of these adaptive elements. So I think the patient perspective can be extremely helpful to design a trial in a meaningful way.
Lola, can you give me your thoughts on this?
Yeah, I mean, I would agree with everything that's been said. I think, even within industry, there is a recognition that patient perspective is really critical to really evaluating these products and being able to describe and qualify the benefit in patients. And I think the agency also shares that perspective. I think the way that the patient perspective is incorporated in the regulatory considerations is around very early on the design of clinical trials, as has already been mentioned, in the selection of endpoints, as an example, whether that's kind of done in a quantitative way where you're directly getting from patients, their experience through patient-reported outcomes as an example, but also patient preference studies as another example, but also in a qualitative way where you could have listening sessions for really rare conditions, rare diseases, where patients are able to share their experiences in a way that informs not only the design, the endpoints, but also how FDA interprets data and contextualizes the data in a benefit-risk assessment. So I couldn't say more about it, and in fact, I think our CRO, as an example, is one that has a chief patient officer precisely for that point, the recognition of the importance of the patient perspective in drug development.
Alan, I'd like to build on that. The other thing that I just recently also found out is that FDA has something called Project Patient Voice, where they're actually asking for a patient to share their daily living and the correlatives that comes with, in addition to the clinical trial data that you're submitting. So, and now whatever information that is not part of the label, you can go on the FDA website and you can find out the correlative studies that are associated with it, that are patient-reported outcomes. And that kind of like helps you, as a parent, it helps me to say, is this going to be tolerable enough for my child or not? And the flux between tolerability and toxicity, how much am I willing to tolerate? What is the toxicity that's coming up? And that perspective is never been present on a label before.
Thank you. One of the challenges of rare diseases is when you do a clinical trial, you may see a site that will see 1 to 5 patients a year. So your trials end up having multiple sites, multiple countries. How do you manage, Chris, the complexity of this, the coordination of this in this scenario?
Yeah, it's not easy. It's something you have to think about in advance. In the example, there's even within a given institution, there's certain specialists. And phase 1 specialists is generally broad. You are used to doing intense PK draws in visits and lab draws, whereas a disease specialty that focuses on phase 3 trials, particularly even community centers, they can't do the intense PK. So site selection is key of what their capabilities, and it may evolve over the trial as well. If you're doing a phase 1 through 3 trial, the sites you need in the beginning of the trial may not be optimal as later. So you really have to have a strategic sense, through the course of maybe you open these 15 sites in the beginning, but you have a goal to get to broader. Back to the patient voice part, you can't open everywhere. We build into our trials travel stipends to move, to allow to support patients traveling to the site, but that's not always convenient for the patients. And then finally, one thing which I've really, on the industry side, grown in a better appreciation than on my academic side, is drug supply and working with the CMC colleagues. Used to just take for granted that drug would be available, when we need it, and really an appreciation of the very long lead times it takes to make drug product through that, and working with your colleagues, and whether it's a pill formulation, an IV formulation, or a cell-based therapy. If you are rapidly accelerating a trial through a seamless design, you don't want to make too much drug at very expensive early on, but if the trial is going well and then you need to rapidly expand, there's a real big time lag. So you really have to work closely with the design and with your broad team about how do you get it, design the right amount of drug and work with your colleagues so that you're not spending or wasting too much money in advance, but you're not running out of drug and inhibiting the seamlessness and having to pause because you're waiting for drug to be made.
Misha, can you comment about that from a patient advocate perspective?
Yeah, so I think one thing that is really important from a patient advocacy perspective is that the access point of view, right? Like there is a drug, but is it accessible? That you kind of said that you're providing stipends, but the other part of it is that the specialty formulation, right? There are kids that have traveled to multiple different sites only to realize that they cannot swallow the pills that are there. So this to me is a question about equity, right? If there's a clinical trial open, if there's an indication that there is present there, is it equitable and accessible to all the age groups? And in this case, if while you're designing a clinical trial, the patient participation is so important. If it is for kids that are under 5 years old, then running through a specialty formulation is absolutely critical so that everybody gets a chance to take. And it also not only helps in access point of view, but it also helps in identifying your sticking to your dosing, as well as your pharmacokinetic measurements as well, if the doses are adhering to a level that you would see in adult population as well.
Narinder, can you give a comment here?
Yeah, so I think as we've been discussing, the seamless trial design and framework does give the opportunity to have to streamline the trial, to streamline costs, efficiency, and many of the challenges that you have when you conduct trials in distinct phases, especially with the startup processes that can take time. But there are, of course, challenges, and it also requires a great deal of to be prepared, because in terms of what the sites are capable of doing, I think as Chris mentioned, we typically sometimes conduct trials in phase 1 sites, and phase 1 sites are used to maybe handling smaller volume of patients, where they will focus specifically on things like dose and adverse events. And now if you're talking about expanding it and maybe having an expansion cohort or even adding on a phase 2 or a phase 3 trial, that might be complicated and prohibitive for a phase 1 unit that's not typically used to handling a large volume of patients. And of course, the corollary is also true that sites that are typically used to conducting larger trials, phase 2 and 3 trials, may not have sort of the capabilities and the infrastructure to handle the intricacies of a phase 1 study. So that needs to be also taken into consideration as well. The other aspect to keep in mind is if you are planning to conduct a seamless trial and you are planning to limit the number of sites because you can use less sites, you do need to keep in mind that you may not be able to get a sufficient number of patients for a given cancer subtype if you're limiting the number of sites, especially for a biomarker-driven patient population. And if you are limiting the number of sites, that also means you may not get a real-world type sampling of the population for a given cancer subtype. So that's also something to keep in mind. Also, I think ensuring that we make sure we have the appropriate US representation for trials, for seamless trials that's consistent with the FDA's MRCT guideline also becomes extremely important. So overall, what I'd say is it takes a great deal of upfront planning in terms of your site capabilities. And we know that in any trial development, things don't go according to plan. Making sure that we have the appropriate contingencies in place, I think also becomes extremely important.
I'm going to go to Lola for this.
I would agree with what everyone said, I think, for the sake of time.
I like it.
Just move on to the next one. Yeah, thanks.
We had mentioned before that flexibility is really important in a seamless trial design, but you need to have pre-specification. I want to talk a little bit more because I think if you don't have that, you have challenges with regulators on, is this endpoint real? Ying, can you comment about that a little further?
Yeah, so everyone like a seamless adaptive design because flexibility. But it's important to remember every adaptation comes with a price. So whenever we do a new adaptation, it's at a chance to make a false positive. And those risks add up pretty quickly. So it's very important to take all those into account and strike a balance between flexibility and statistical and regulatory credibility. And the best way to do it, as I mentioned, is through computer simulation. Because we don't know what kind of interim data we're going to obtain. And the decision will depend on the data. So that means the best approach we can do is to simulate all the possible cases and look at all the possible adaptation we may have. And through the simulation to demonstrate the design is robust enough, performance very well under all the scenarios. And for those design, especially those complex design, regulatory, like including FDA, typically will require extensive simulation to demonstrate operating characteristic in the submission.
Thank you. Any other comments on this topic in this room?
I could just add that I think, as I stated before, I think it's really important to determine a priori where the key decision points are and what the critical scientific questions that are intended to be answered during the course of the clinical development program, because I think sometimes the risk of that I see with the implementation of these seamless designs comes in sort of reacting in almost real time to information data that's being collected. And I think when you're doing that without a plan, you obviously risk introducing a lot of multiplicity issues in the overall investigational plan. And then what you end up with is sort of a case series, right, where you're describing data, but you're not really able to really quantify, benefit or reach some definitive conclusion. So I do think that it's important to be succinct in the number of questions that are being asked of the study. Every study can't answer every question, and I think in the rare cancer space or rare disease space, there is a tendency to really, like, this is our one shot. We're going to answer all the questions that ever, were thought of. And I think we run the risk there of reaching false conclusions when we do that.
Thank you. FDA is quite important in this process, but they're not the only regulators. It's often more challenging to balance both the FDA and EMA, et cetera. Harinder, can you give me some examples of best practices you use to try to coordinate these efforts?
Yeah, so as Lola mentioned, seamless trials are iterative, right? So as you generate data, as you learn about, how the patients are responding, as you learn about the drug, what it allows you to do is apply some of these adaptive elements so that you can refine the trial, you can make necessary changes to optimize the trial, to reach your objective. And, but that also requires a very fine balance between pre-specifying so that you don't introduce any kind of bias and you preserve integrity, but also making adjustments with some of those adaptive elements. So I think it's extremely important to have engagement with FDA to ensure that there is alignment. With respect to the US, you can leverage the FDA formal engagement, such as the types B, C, and D meetings to get agency guidance. So you can envision a scenario that when you're designing a seamless trial, you have a type C meeting where you discuss your overall trial design with the agency, get their guidance, get their input, and then once you embark on the trial and you start generating data, you have to make some adjustments to some of those adaptive elements. You could use a type D meeting, and the type D meetings usually you can secure quite quickly and get those sort of check-ins to get the appropriate guidance from the FDA and ensure that you're on the right track. But Ellen, also you mentioned that we all conduct global trials, global multi-regional, multi-site trials, so it's also extremely important to have discussions with the agencies internationally as well, because we know there may be differences and divergences in the regulations around standard of care, how they define patients, data collection requirements, things like whether or not they will accept the flexibility in making modifications to the trial. And then we also know with things like EU CTR in Europe, there are certain lead times with making amendments to trials as well. So it is complex, but it's also an opportunity, and so I think ensuring that we have dialogue with agencies around the world so that we can come to a common framework that can ultimately deliver our objectives on the trial, I think becomes extremely important.
Thank you. Lola, what are your thoughts on doing that?
Yeah, I mean, I think at the risk of sounding repetitive, I think, there's obviously always an urgency in the rare cancer space, right, to develop the product and to do it very quickly, efficiently. I do think it's really important to take a step back and really put in the necessary time to develop that comprehensive strategy from beginning to approval and to post-approval. And I think having those conversations around the approach to doing that as early as possible with the regulatory bodies is critical. Even when you're not in the clinic, there are opportunities to engage with the regulators and to put forth that plan. And so really leveraging every opportunity where there's contact to define and continue to propose that plan is important to leverage.
Thank you. Looking ahead, what do you think could help expand the use of seamless trial design and strengthen rare cancer development? And for this one, I'm going to start with Chris, get his perspective.
Yeah, so I think to build upon that last topic is just engagement with global regulatory bodies to get them to buy into this, but also to support and understand level of evidence. It really is a balance between scientific rigor, but also practicality in moving things forward. And kind of similar to the last topic or the last question, the world is not aligned on that. I think we've been fortunate in the U.S. To try to move things forward, and I think the FDA has really tried to be responsive to some of this. I've not necessarily seen that in the rest of the world as much, where we've tried to be brought trials down to the age of 12 and not just 18 for an adult population based on the scientific evidence and preclinical data, but I've had other regulatory bodies outside the US say, well, until you show evidence in adults, it's unethical to study in an adolescent. And so I think working with the global regulators to really understand the need and the urgency, but with scientific rigor, I think we have a ways to go there.
Yang, can you give me your closing thoughts, please?
Yeah, to effectively use those seamless design, I think one barrier is we need enough experience, both how to design those trials and how to operate those trials. Build a robust infrastructure to allow us to collect the data in real time, in high quality, to facilitate those seamless and adaptive decision. That's really important. At MD Anderson, we are doing quite a bit adaptive design. And even though still we need a lot of infrastructure and experience to do more such seamless design, especially for rare disease, for which power is typically very limited. And it's very important to have efficient design and implement it in a perfect way so we can make it successful.
Thanks. Lola?
I think, certainly in the working groups, we didn't invent anything, and I don't think anyone here would suggest that we came up with something, we learned something that had not been, where there was no precedent. And so I do think that there are, what was very clear in our working group was that, discussing specific examples really enriched the discussion, and it helped us to really identify and name what you were doing as part of this adaptation or seamless study design. So I would say that, what could move things forward would be more collaboration and sharing of real cases, and more transparency about what worked and what didn't work, what was acceptable and what wasn't acceptable, and why, would really help to move this approach forward more.
Thank you. Harindra?
Yeah, look, I'm really excited about the opportunity and the promise that actually a seamless trial design provides. Ultimately, it's about, I think, Friends of Cancer, and I think all of us is about getting novel therapies to patients as soon as possible. And I think seamless trials has the opportunity to be able to deliver on that. But as with any new initiative, I think there are going to be some learning experiences along the way. I know companies have been doing some forms of seamless trials, and I think this is a great start to sort of pull everything together and provide that framework. But as was mentioned, I think the only way that we can all, move this forward is by sharing examples of what worked, what didn't work, so that we can continue to optimize and come out, with what we think is the best trial design moving forward to, again, enable early access for patients.
Misha, I'd like to hear your thoughts.
I think what everybody's already said, right, that all of that makes sense, but I think to the core of this has to be patient-centeredness. We have to have patients involved in those conversations before the protocols are finalized, right? It should not be an afterthought that the protocols are finalized and we are going to do X, Y, and Z, and now you bring into the patient and then you realize that this does not work, right? Because this is not what is evidential, or it's affecting the quality of life of the patients going forward. So you might not get retention and stuff. But I would like, everybody here to even think about in a situation where there is no standard of care, right? In a disease where there is no standard of care, the urgency is great, the unmet need is even significant. Do these regulations or do these considerations even play in, say that you need to wait and have a precedent? That's a question that it's for everybody, right? Do we need to have that, or do we just march forward and be the first one to do that?
And before we go to questions, I'll just kind of close. I think part of it is changing your mindset, thinking that we can do this quicker and finding a way to move forward for patients. If you have the patient as your North Star, I think that's critical. It's also important to ask. FDA was very clear, come talk to us, which, You may not think it'd be helpful initially. It is. They're very open to it, finding new ways, and it may take a couple conversations, which may not sound the best from a company perspective, but it's the right approach if you can accelerate and get drugs with the ultimate goal, get into patients as quickly as you can. I think we'll go to questions now, unless there's any other comments on the panel. I put on my glasses now. What is the difference between seamless and adaptive trials? People often use them interchangeably when speaking. Should we harmonize that, or is there a difference of true importance? Yes. I'll start to say it's a word that people use, and it depends. There isn't a right approach. Some can be seamless, some can be adaptive. It all depends on how you're designing the trials for the endpoint. For example, if this is a phase 1/2, it may not be adaptive. It may just be looking for the right dose selection. But I'm going to go to Chris to address it a little further.
Yeah, no, I think you just hit upon it right there, is that, there's overlap. You can be seamless and adaptive. You could be seamless, or you could just be non-seamless and be adaptive. All of them apply. It depends on your end. Adaptive ultimately refers to the endpoints, and that you adapt along the way. You're changing along the way, whereas seamless is really just moving along the paradigm from first in human through a regulatory endpoint or somewhere in between. And you don't have to change, to your point, the endpoints. You can have the same endpoint along the way, and do that. So they are separate concepts. They can be combined together, but they do not have to be.
And I'm going to get Ying to comment about the statistical aspects of adaptive, because I think that has a different meaning to me when you say adaptive.
Yeah, strictly speaking, a seamless design can be viewed as one type of adaptive design. But most seamless design will involve adaptation, because the basic idea of a seamless design is you combine different phases. And in one phase, you learn something that will be informed how you do the second phase, right? We do phase 1, which is going to determine the dose for phase 2. And that's, if you look at the whole trial, that's adaptation. It's not only seamless, but also make an adaptation in terms of dosage, in terms of population, in terms of even endpoint. So in some sense, they are so closely related. That's why I think in our conversation and also a white paper, we kind of use them in an exchangeable way. But I think in our setting, maybe it's not that important to distinguish seamless design and adaptive design. And in a statistical sense, what happens is you're going to use the data to inform in-trial decision. And that will impact the operating characteristic, statistical property of a whole design. So that's why we put them all together, take into consideration, adjust for those effects to ensure study integrity, like strictly control false positive rate.
Yeah, I mean, I could add that we were intentional about not providing sort of, strict definitions on characterizing, an adaptive trial versus a seamless trial, and there's a great deal of overlap. And I think having that flexibility, I think, is important because, as was mentioned, we typically conduct trials that are fit for purpose, for a specific disease, for a specific patient population. And in terms of whether you can apply a seamless framework, components of a seamless framework or an adaptive needs to be determined kind of based on what you're studying, the patient population. So I think there'll be various iterations along the way, and I think that's okay. And it'll certainly contribute towards, I think, designs that we can learn from and optimize, as we develop drugs.
How does a seamless design deal with the recognition of new toxicities that appear after phase 1 and more patients enrolled in subsequent phase 2 or 3 design? And I'm going to pause, and then I'm going to go to Chris, I think, first. I think some of it depends on what you know about your molecule and what trial design to do. If you have a drug where you're really going towards MTD, you're going towards a pretty toxic level, you may be doing more of a phase 1/2 rather than right into the phase 3. So it depends on your drug itself. If you have a drug that you think is a little more tolerable, you might do more changes. But there are always changes that happen. There's toxicity that happen, and maybe Chris can comment what happens when you do see these side effects in a late stage development.
Yeah, I think it's trying to understand too early toxicity versus late toxicity, and whether it's seamless or not, you're always using earlier data to inform later. So even if you weren't doing a seamless design, you may still be following patients from your phase 1 as you're running your phase 3 and learning new things. You build as much as you can into the beginning and be thoughtful about it. As with all trials, you sort of have dose modification guidelines that are both specific but also that cover broad. And the one thing, the other aspect and the reality of it is there's amendments. No one likes to amend trial over and over again. They're problematic, they're expensive, but it's the reality, it's what happens. And so I think to answer this question is if you're learning new information that you hadn't thought of, you thoughtfully work that in, and you can potentially amend the trial later. There are challenges with that, but it can be done.
And I would say that there should always be a plan to look at late toxicities in any development program. That should always be sort of considered. You have to think about, the information which, from the early part of the development program is around more acute toxicities, right? And so I think there should always be a plan. Whether you know the molecule or you think you know the molecule, I think we all know that there can be some surprises there. And so there should always be a plan to look at the totality of data, throughout the development program.
Any other comments?
Yeah, so in this case, it may be difficult to integrate phase 1 data with later phase data. It's one of the goals of seamless design, because we want to maximize the information. We want to aggregate all the data from beginning to end. So in this setting, it may be a challenge, and most likely we will focus on phase 2 or 3 component to inform a final conclusion, because it's different dose. But still, seamless design are still efficient in the sense it's eliminated all the white space between a trial in the conventional way. So from that perspective, it's still efficient and useful.
Thank you. How do you handle blinding in a seamless randomized phase 2-3 design when the intent is to carry out one of the arms from phase 2 compared to the standard of care in a blinded phase 3 study? Ooh, that's a tough one. I think one of the challenges you also run into, and I've been in a small company when you have a blinded trial, what do you do when you have a safety signal and you don't want to unblind, especially in a small company? I know what I've done in the past is I've had external people who are unblinded to look at safety from an unblinded fashion. In a larger company, you have complete divisions who can do this in an unblinded fashion. We've not talked about this from a seamless trial design, but maybe Harinder can comment on that further.
Yeah, so actually, even if you think about the Project Frontrunner concept, which was presented recently, it sort of has the same idea where you sort of take an early look at your phase 3 trial data, and you have the potential if you meet that primary endpoint that you set for that early look, whether it be ORR or some other surrogate endpoint, that you can file for an accelerated approval, but you continue your trial maybe in a so-called blinded way until you get to your final endpoint. And in those types of situations, you can do things like what you mentioned, like sequestering, where you have sort of different groups looking at different parts of the data and doing the analysis so that you kind of preserve the trial integrity. So there are, I think, creative ways that you can come up with to preserve the integrity of the trial that you can also apply to into a seamless trial. As you said, we didn't get into sort of the nuts and bolts of how we would do that, but I think there are probably ways that you can work through that to find some viable solutions.
Any other comments on this question?
Yeah, one way we can maximize the blinding integrity is we can utilize external committee, interim decision committee or dose selection committee, and who can access unblinded data and make recommendation which arm should move forward. So in that case, you still maintain most of blinding, right? You have arm A, B, C, and eventually the committee recommend B and C continue. But you still don't know B and C which treatment represented. So in that case, we still can maintain blinding.
I can give a practical example of that because a phase 3 trial is ongoing in a rare patient population. It does have 2 doses. And as a smaller company prior to my recent one, we did have the IDMC chair be in charge of looking at the safety in comparison between 2 arms and have them as a responsibility to see if there is a signal for an issue. We would only be unblinded if we need to make modifications. So there's one way you can do that. You do give up control, which no one likes to do, but I do think in a scenario where you're trying to find what's the right approach for patients, you can do that approach. I think the last question is what are the next steps in moving forward in a seamless clinical trial design? And I'll just preface this, it really depends on the molecule too. If you got a very early phase drug where you're not sure the safety, you may have to do more of a dose optimizations phase 1, more like the FGFR one, where you're looking to find what's the right optimal dose. There isn't one right approach. You do have to understand your drug. You have to understand the disease setting, the unmet need, and how to move forward. Someone else on the panel can address that? Chris?
I think, as we talked through our number of examples, where it's been successful to date has been a lot of biomarker-driven disease, particularly small molecule TKI type of drug development. That's where there's been a number of successful examples. I would love to challenge all of us of how to move beyond that and use it in different settings as well. Take the models there and think about ways. But I think a lot of it depends on having strong preclinical data or preclinical correlating to clinical. And from a company perspective, back to the blinding question, like if you're going to do seamless and invest in multiple phases at one time, you have to have conviction that there and put money into where that is. And that's a lot of risk. So you have to have decent data. And in the small molecule space, that's a little bit easier. It gets challenging in other therapeutic indications and other types of molecules. Immunotherapy is a little bit more challenging and would love to see it broaden.
Lola, go ahead.
Yeah, I would just, again, sort of emphasize that I think the approach to the clinical development program, including specific trials, really should be based upon the questions that are being, asked. So a seamless approach may not be appropriate in every setting because there's a lot of reliance on assumptions, and I think one has to consider what are the data that are informing those assumptions, and what are the risks that people are willing to take, whether it's risks from the safety perspective, risk of failure, financial risk, and other kinds of considerations. And so I think what is really the driver for any approach is what are the questions being asked and what do we know a priori before we go into it. So it may not be that every clinical setting is appropriate to take on this approach.
I was just going to give a sort of direct answer to the question is we should just go for it. I think particularly with rare cancers and ultra-rare cancers, there is support that this design has a lot of upside, I think, to bring therapies for patients. And the only way we are going to learn is by actually trying it. So I would say if you have a design that you are considering it, talk to the agency, talk to health agencies, get their guidance, and try it.
Well, thank you, everybody. You've been a great panel. I really appreciate your participation. And the job you've done getting this white paper to where it is. So thanks again, and I appreciate the opportunity to talk.
Thank you very much to this first session for getting us started with a great opening discussion. Thanks for everyone for your contributions online. We had a lot of questions coming in. We will do our best to get to as many of those throughout the subsequent discussions and find ways to address those moving forward. I do want to take just a minute to highlight a new research partnership that Friends of Cancer Research has launched that's related to the topic discussed earlier today. This is related to the use of external data to supplement clinical trials, an approach that could be informative for evaluating drugs for rare cancers. In our new external control arm pilot project, or our ECA pilot, we're working with 8 different data partners that will be constructing an individual ECA by matching patient characteristics of control arms of the control arm of a recently completed clinical trial, each using their own data. From this project, we'll have the opportunity to evaluate consistency between the traditional control arm and the ECAs and begin to identify characteristics of the source data which may contribute to any of the observed variability. Ultimately, we hope that this will be able to pinpoint the necessary data to successfully construct an ECA as a potential tool in drug development programs and help contextualize future results. So stay tuned as this project progresses. Our plan is to discuss this and the preliminary findings from this project in 2026 at a meeting scheduled currently for April 7th. So we are now going to take a break between our morning sessions. Please take the opportunity to visit with colleagues, refresh your coffee out into the foyer. And just as a reminder, we do have a large online contingency today, so we're going to do our best to stick to the times listed in the agenda this morning. So with that in mind, please take the opportunity to stretch your legs and be back for our next session at 11:40. At Merck. Gideon? We'll get you all. Go ahead and grab seats. Yeah, thank you.
Okay, thanks so much, Jeff, and great to be here. Thank you to Friends of Cancer Research, Ellen, Jeff, for inviting us to participate in this great meeting. Also, thanks to Hillary Andrews for shepherding us through the working group. It's a little unfortunate that as a former Fed, our federal colleagues couldn't be here. They worked so hard when participating in the panel, but we have a great panel, and we'll try to do it this topic, this very important topic of trial designs for combination drug development justice. So. Combination therapies in cancer care. This is a crucial topic because in cancer, dating back to the origins of oncology, I saw Bruce Johnson is in the crowd. There he is, Bruce. Dating back, from the original chemotherapy regimens, this has been an ongoing topic. How do we best combine chemotherapy regimens and assign a contribution of components, kind of moving towards the model that infectious disease uses in HIV and tuberculosis. So drugs can combine in a few ways. They can be synergistic. That can be controversial. Some people don't believe in synergy, but I think there have been examples in oncology where synergy does occur, where the combination results in greater efficacy than the individual components. There's also additive, 1 + 1 = 2. Very simple math. The contribution of components equals the sum of the effects. And then, you can see unfavorable if you have overlapping toxicity and patients have to discontinue treatment early. That can be an unfavorable combination. So assessing the contribution of effect to discern the individual contribution of each component is critical. And we all know that factorial designs, there are limitations. Scientifically, it's the best approach. But we live in the real world. We have to be practical, and we can't test every single component individually. So treatment A, treatment B, treatment A plus B versus the standard of care. And the infeasibility could be due to increased complexity of the trial design when treatments are used. The number of patients can be prohibitive, and there are ethical challenges. Sometimes, treating with a single agent may not be ethical or patient-friendly approach. So the practical design considerations for alternate approaches to factorial trials. In this working group, we explored other considerations for trials utilizing combination therapies, and we used case studies to determine what evidence is sufficient to demonstrate contribution of effect. So this could be clinical data. This could be data modeling and non-clinical pharmacology. Luckily, we have Julie Bullock, who's a pharmacologist, to help us through some of the modeling and pharmacology aspects. And you can leverage data from other settings. So the objectives, this panel will discuss considerations for designing clinical trials, utilizing combination therapies, highlighting areas where additional clarity, flexibility, or alternative approaches may help to operationalize these principles. And we'll examine scenarios where alternate designs may be useful to obtain regulatory approval. So we will now go forward with the panel. So why don't we kind of just go down the road, just introduce yourself, get a little bit about your background before we start.
Sure. Thanks, Gideon. So hi, everyone. I'm Julie Bullock. I'm currently an early development lead at GSK, and previously to GSK, I was in consulting, and I was also at the FDA as a clinical pharmacology reviewer.
Hi, everyone. I'm Gary Doherty, representing AstraZeneca today. I'm a medical oncologist, previously in Cambridge, before moving to AstraZeneca 3 years ago, where I lead one of our immunotherapy programs and lung cancer and head and neck strategy.
My name is Elad Sharon. I'm a medical oncologist at Dana-Farber Cancer Institute and an associate professor of medicine at Harvard Medical School. I've been there now for a couple of years. Prior to that, I was actually working on immunotherapy, largely immunotherapy drug development for the National Cancer Institute at the Cancer Therapy Evaluation Program on early to late stage trials.
And I'm Carol Villette. I'm an advisory advocate for the Friends of Cancer Research, also a Komen advocate in science. I've been treated for breast cancer in 2008, I think at this point. I think that was the year. And I've been involved with the Friends of Cancer Research for, I think, about 3 or 4 years now.
Okay, great. And just as a reminder, just like in the last session, we'll have some time for Q&A. At the end, so you can scan the QR code on the agenda or click the link dropped into the chat to ask a question. I think we already have a host of questions already, so fear not, we're well populated with questions. So to start off the panel, what are some practical and scientific challenges with factorial designs that are leading sponsors to consider alternative designs or making those alternatives feel necessary? Why don't we go with Gary first?
Sure. Thank you. I think we all recognize the importance of demonstrating contribution of effect. And we've had some very robust discussions in the group from many different perspectives where we've really tried to reach consensus, where we're trying to balance the methodological rigor and the need to have this scientific explanation and quantification of contribution of effect with the practicalities of drug development. So we see with full factorial designs like the one that you have presented, Gideon, in the presentation earlier, where we have the combination treatment, where we have 2 arms for each of the monotherapies and then a standard of care arm. that can lead to really very large studies with very long timelines that ultimately leads to potential delays in patient access and approvals. And what we're trying to balance in this white paper is the need for that methodological rigor and the data package that can support contribution of effect and approval with those practicalities of drug development. So there are important feasibility issues, not just the ability to enroll the number of patients that might be needed in a full factorial design, but also making sure that each of the arms in that design is something that makes sense for a patient from a clinical point of view. And we've been discussing a lot about monotherapies, and sometimes the monotherapy could be poorly effective and may not be something that we would want a patient to be randomized to versus the standard of care. So there are lots of these individual practicalities that can, they're not just additive, they can compound each other, that lead us to investigate other approaches for demonstrating contribution of effect in our drug development programs. And we've discussed in the white paper, and we'll discuss today, some of those different approaches.
Yep. Thank you. And Elad, you moved from government, from CTEP to a small hospital, teaching hospital in Boston. So yeah, what are your thoughts on these challenges, from a clinical perspective?
Yeah, I have to say that, of course, preclinical experiments are often used to actually guide what we do, especially in the initial early phase of clinical trials. And frequently we see and have seen that combinations are more effective, obviously, in the preclinical sphere. And so what we need for the most part in the clinic is some sort of real proof evidence that the monotherapy of, let's say, what presumably would be an additive agent, is ineffective. We don't, just like Gary said, we don't necessarily want to actually randomize a patient to a wholly ineffective drug, but we do have to actually demonstrate that in the clinic and not just in mice. And there are ways to get around that. In terms of trial design, one could actually design, let's say, a monotherapy arm that has crossover at the point of progression to allow patients to get access to a new drug after we've proven that monotherapy is ineffective in some manner. But that's critical, and it's critical down the line, because ultimately, when we have combination clinical trials, we have a situation in which patients are potentially exposed to more agent, therefore more toxicity. And that has to be worth something. We have to know what that value is for the individual patient. And in order for that to be clear, we have to actually demonstrate ineffectiveness, or at least poor effectiveness of a monotherapy that we're considering in combination.
Thank you. So, and Carol, from a patient advocacy perspective, what are your thoughts here?
Well, we all know that combination trials with many arms require a far greater number of participants. And it can be really a tough job to recruit the number of patients needed for a trial like that. So it's absolutely necessary that patients who are asked to participate in a trial understand the risks and the rewards to them, possible rewards to them, and how the study might bring some toxicities, but also could help others in the near future. And the more complex a trial is, the more this needs to be communicated. When patients sign up for a trial or agree to be in it, it needs to be very, very clear to them. And I think it becomes, with too many arms, just a complex process, and people become confused. I know when I participated in a few trials, it's a tough decision. People don't willingly become, and I still hear this term from patients, guinea pigs. And we don't want them to feel that way. So alternative trial designs can help lower that burden on patients and help ensure that we do have good science that will, in a streamlined manner and in a perhaps a quicker manner, develop therapies that are effective with minimal toxicities for patients, and that we can have great progress in this area.
All right, thank you. So we know that there can be cases where a sponsor can justify not including the monotherapy in the arm. We have one example in the white paper of KRAS G12C inhibitors in colorectal cancer in combination with EGFR monoclonal antibodies, which have boxed warnings saying you shouldn't use EGFR mAbs in KRAS mutant colorectal cancer. But yet, the combination, when you combine it with a KRAS G12C, it works quite well. Another example, would be what we learned with BRAF MEK inhibitors, like BRAF on its own, although quite effective in melanoma, can cause this skin toxicity, keratoacanthomas, where if you add in a MEK inhibitor, it can mitigate the risk of this toxicity. So I don't know, Gary, do you have any thoughts on, from a sponsor's perspective, when do you think it's okay to forego the monotherapy experiment?
I think it's very situation dependent. I do think it depends fundamentally on the mechanism of action of each of the individual drugs, what we're expecting in terms of the combination, and although synergy is a potential dream, it is a reality in certain situations, and it's something that we hope for in drug development combinations. So I think preclinical data can certainly support this. There are instances where you just simply wouldn't expect monotherapy activity, or where you expect it to be poorly effective, and where the other agent is not just carrying all of the effect in the combination, but where they act together in a way that we can describe mechanistically, that we can have preclinical experiments where this plays out, and then the early clinical data, depending on how that's informed by the preclinical work, can further support that, leading us to omitting those monotherapy arms that may not be attractive or clinically sensible for patients to enroll in.
Great, thank you. And Carol, as a patient, obviously, one wouldn't want to be randomized to an ineffective treatment. So yeah, how do you kind of advocate?
You're exactly right, that if the data clearly show an arm is ineffective, it wouldn't make any sense, nor does it wouldn't be ethical at all to randomize patients into a known ineffective arm and expose them unnecessarily to toxicities. And I don't think anyone would want to recruit people into a study like that. It just wouldn't make any sense. So I agree completely that it's just not something that we would want to do. Yeah.
Great. So moving on to data to support alternative trials. Our white paper highlights the necessity to have data to support COE before registrational trials. How should sponsors be thinking about preclinical and early phase trial designs to support this? So Julie, what are your thoughts?
Yeah,
so early drug developer.
Early drug developer. Yeah, I definitely think that there is a lot of opportunity for early phase study designs to not only establish POC, which is something that we all want to do in early drug development, but provide those early pieces for contribution of components. And I think that that's usually where a lot of sponsors have a tendency to we're trying to accelerate things as much as we can, and so it's easy to kind of leave the contribution of component exercise until the registrational study, but there's quite a bit of benefit that can be gleaned from including some of these exercises in early studies. One is it de-risks the entire combination program, right? You're less likely to take a combination into phase 3 that may not prove to be, a positive contribution of component. And then the other is that it could potentially come in helpful when you're trying to decide, what arms in my factorial design do I want to drop? And so having that internally compared data in an early phase 1 study is definitely very, very helpful. And I think having early foresight to build that in is something that a lot of companies should be doing if they think that this asset is potentially headed towards a phase 3 combination trial. We had a lot of very animated discussions in the working group meetings with regards to inactivity, and then it's like, well, then what is activity versus efficacy? As a pharmacologist, I usually think of activity as something that's a dynamic marker or a mechanistic marker of, we're hitting our target. or potentially biomarkers, depending upon how relevant those are to the mechanism of action as well. And those sometimes are correlated to efficacy, but sometimes they're also not. I mean, if we could approve drugs on pharmacology alone, there would be a lot, drug development would be way simpler, you know, but sometimes just hitting your target and showing even a dose response or an exposure response isn't usually enough to get us to an efficacy threshold, whether that's a response rate or PFS or, you know, eventually OS. So I think that, you know, having some understanding across a dose range of how your activity is changing when you're in combination is incredibly insightful to help you hopefully translate to efficacy later down the road. But activity alone is not, I think, enough to really be the end game for a contribution of component exercise. I really do think we need to have efficacy.
Yeah, good point. And like as Dr. Pazder likes to point out, you know, with ipilimumab, you know, there wasn't a lot of single agent activity, but, you know, if you only looked at the response rate, you might throw away the drug. So you also have to look at other factors like durability of response and other things. So Gary, moving from early stage to late stage, you know, what are you looking for, you know, when your early stage colleagues hand you, you know, throw it over the fence to the late stage and want you to think about, you know, proof of concept and you know, you're moving towards registration. What are other factors you consider?
I consider why we have a fence. So I think increasingly we need to see contribution of effect as a program level decision and an evidence package and not a single study output. And I would be looking right through from preclinical to the early clinical data. So the preclinical data might support that the two combination monotherapies have distinct mechanisms of action, that they have non-overlapping resistance mechanisms, for example. I think there's a totality of data there that is not in itself sufficient, but it can support the early clinical data. And it's great whenever the early clinical data and the preclinical data are concordant, where everything's pointing in the right direction, where you have a package and a story that makes sense, it's evidenced, and the totality of the data can then inform the optimal phase 3 design. In terms of the early clinical data, so we obviously have reliance on clinical endpoints like response rate, duration of response, PFS. But increasingly, I think we need to think more innovatively and think about, you know, the potential for other endpoints in these studies. For example, PSA in the prostate cancer setting, ctDNA, multiple tumor types now. And I'd love it if we have a totality of package of data that creates a package that we can take to our regulatory colleagues, and where we can really streamline the later phase development for the issues, given the issues that I've outlined earlier.
Great, thanks. And Alad, in your role as kind of the guru of toxicity, how do you consider when you're combining two agents and understanding the contribution of each agent in a combination from a safety standpoint? How does that work?
No, well, certainly. So safety is an obligation throughout the clinical trials and actually even, you know, beyond. And obviously rare things actually don't even get picked up on these early small trials. So there are you know, certain limitations. But toxicity always has to be looked at in context. And so if there's a certain efficacy threshold that's met, then you know, perhaps we're actually able to handle the toxicity or at least mitigate and deal with. And that's obviously, you know, one of our goals. You know, in terms of you know, clinical trials, thinking about the design, just going back on your ipilimumab example, I'm just reminded, and I hope the story is correct. It's an apocryphal story that I got from someone, I think, at the FDA, that suggested that initial clinical trial looking at ipilimumab at a 10 milligram per kilogram dose plus GP100 vaccine, which was, I think, the original Steve Hody trial that was published in 2010 in the New England Journal, was initially only going to be compared to the vaccine, to the GP100 vaccine alone. And it was actually the FDA that actually pushed back and suggested that there would be another ipilimumab arm, ipilimumab by itself, that showed the benefit to IPI, but rather not to the vaccine itself. Both in that trial, the only agents that were offered to patients were actually all investigational. The GB100 vaccine and the and ipilimumab that led to the approval of ipilimumab. But it was that design decision that basically turned ipilimumab specifically into something that was more generalizable and not something that could only be used with a vaccine that was unique to melanoma. Because the GB100 on its own did not really show significant activity, and no real difference in the combination versus IPI by itself. They beat, obviously, historical controls, and that's what led to that initial approval for ipilimumab. So sometimes we find this out, obviously, in late stages, despite the best of our abilities or interests or plans. But clearly, that contribution of effect had an impact on later development in immuno-oncology in general, and it was the foresight of the FDA that got us in the right direction. At least, again, that's what I tell my fellows, so I hope it's true.
I thought I saw Pat Keegan in the audience somewhere, so I think he can confirm, but maybe not. But yeah, that's a great point. So. During our working group calls, we discussed the potential for performing non-powered analyses, and we don't have a statistician, so we'll have to all,
pretend to be one.
Yeah. Okay. But what are some opportunities and challenges of this approach for these non-powered analyses, Julie?
Yeah, so I'll go ahead and start on this one. Yeah, this was another spirited debate amongst the group, and I know that the FDA had a lot of thoughts on this as well, as they should. But I think that when it comes to non-powered analyses, it really comes down a little bit to the magnitude or what you're trying to conclude from the non-powered analyses. Are you trying to conclude that a monotherapy arm isn't effective? Are you trying to conclude that there's no activity so that you don't want to randomize patients to that? I think a lot of it comes down to not only the descriptiveness and how, the totality of data from like the PK, the PD, some of the other biomarker data that we have that go into it, but it also comes down a lot to sample size, right? So it's like a non-powered analysis of 6 patients in 2 different cohorts is probably not going to be enough to contribute anything from a meaningful conclusion, but potentially, we make a lot of decisions actually in drug development based on non-powered analyses. I mean, the entire early development space is typically a non-powered analysis exercise, and we make pretty big decisions on non-powered analyses there. So I think it can be done, but I think we really have to make sure that we have agreement with what we're going to be using it for, and what working with the agency to make sure that we have enough robustness around the non-poweredness of these designs. Yeah.
Yeah, and I think, we have some examples. For example, PD-1 plus CTLA-4, where, originally in melanoma, it showed superiority to CTLA-4 alone. And then I think over time, there was increased confidence across different tumor types that, there seemed to be, effects, across tumor types.
Right.
So you're kind of leveraging data
across different tumor types. And then I also think we're not talking about it quite so much today, but we did talk about it in the white paper, the concept of using real world data or external control arms too. I think that's also another issue when we were talking about non-powered analyses is it in study or is it coming from an external source? And the power of descriptive statistics when it comes to things like that can be a little bit questionable versus stuff that's in a controlled study where you can actually do a good comparison. Yeah.
And Carol, what are your thoughts?
Well, I agree that in some cases there's good reasons to conduct a non-powered study. And I just have to share this example, and my friends here can correct me. When I was preparing for this, I said to my husband, who's an accomplished engineer, I said, what do you think about a non-powered study? Would you accept results from that? And he said, what do you mean? What does that even mean? And so I explained, there's a difference between a large number which is not, but still not statistically significant as opposed to a small number. He goes, oh yeah, why don't you just describe it that way? And I think that goes down to the heart of how patients would feel about it, that if it's a tiny number of people, like 5, we're not going to make decisions based on that, as Julie said. But if it's a large number of patients who've been in a study, it looks like there's a positive outcome, even though it's not powered to a statistical level. It's something that patients can and their caregivers can understand, because, I believe that patients and their families are very savvy, especially with online resources now, and they will understand that. They will understand the difference and will make decisions accordingly. So I think that it's certainly something to consider, that you can draw conclusions from what's considered a study that's not sufficiently powered.
Yep, and it's a great point that you and your husband make. I mean, I think a lot of times we get bogged down in the jargon, but to really be precise to patients exactly what we mean when we say these things is really important. And Julie, you kind of stole our thunder, because we are going to talk about real-world evidence.
We are?
Yeah.
Well, I'm not talking on that, but
Okay. Well, feel free to chime in.
I might have to.
So, so how might external real-world data evidence, preclinical data, or modeling, I think you should chime in on this, be considered to support COE? So we did have one example in the working group where we talked about using real-world evidence from a large registry in France in lung cancer, whereby we looked at BRAF mutations, and we saw that it wasn't prognostic, and that helped, with our review of a BRAF and MEK inhibitor application for non-small cell lung cancer. So Gary, what are your thoughts about novel combinations leveraging real-world evidence?
I think it depends on the definition of a novel combination. If it's truly novel, then these drugs as monotherapies are not approved, so the ability to collect real-world evidence won't be there. I think if we have a drug that is approved, for example, for a different indication where there's off-label use and there's a sufficient body of evidence that can be collected in the relevant disease setting, then that certainly could be relevant. It's something that we were focusing mainly in the working group on novel combinations, but you can imagine scenarios where a contribution of component arm could also be an acceptable standard of care for that population, but is not the control arm for the study, and real-world evidence could come into play there as well.
It sounds like Friends is working on a project on external control arms as a potential synthetic arm. Yep. And Julie, from a modeling and simulation standpoint?
Yeah, so, I mean, I think that there is, a lot of it depends, like Gary said, on what you're trying to get out of this external control arm. And I think one of the largest problems that I always had when I was in consulting working with clients on this is that, well, we're going to use real-world data, but we're developing our drug in a biomarker subset. And I'm like, okay, does the real-world data have the biomarker subset in it? Like, that's like a really key thing that people sometimes just forget when they're thinking about leveraging a registry or other items, is that, this mechanistic marker that you're going to develop the drug in may not be in that real-world data set. But for more like, established standards of care or established assets that already are approved, we have, and the agency has even used, MBMA, model-based meta-analysis, to help provide context for single-arm trials, right, back in the day when those were a little more common than they are today. But we did use model-based meta-analysis, which is basically just accumulating data from the literature and other public sources to basically create like almost a synthetic like control arm in a way. And companies use these model-based meta-analysis a lot of the times internally to decide whether or not a drug is going to move to phase 3. They'll do model-based meta-analysis to sort of benchmark themselves. And so I think that for the concept of combinations, like maybe we could use this for like the standard of care arm, And it could potentially be used for contribution of component exercise, but again, it just goes down to the quality of the data. And that's usually, as former regulators, we always struggled with when it came to real world evidence and whether or not it was going to be acceptable for regulatory purposes. Yeah.
Yep, indeed, and it's a matter of, having high quality data and then using propensity score matching, and
Right.
by the time you funnel all that data in, it becomes smaller and smaller numbers.
Yeah. With regards to like modeling, I mean, and this is one of my things for the last question, but I'll maybe touch on it twice because I think it really is important. With the concept of Project Optimus, I'm hopeful that the drug development paradigm and landscape and combination development will become a little more rigorous in early development. And so, with that rigorous dose optimization exercise that we are hopefully going to be doing in the future for combinations, we should be able to leverage exposure response to hopefully help with contribution of components. Because if you see an exposure response across different dose ranges in combination, that basically is sort of like an internal control for your contribution of component exercise. So I do hope to see more of that in the future. I don't think that we've seen a lot of it to date, because we just didn't collect that level of data in combination historically in oncology. But in the future, hopefully we can.
Yeah, and hopefully, maybe this is a subject of another white paper, but
Right.
How to extrapolate combinations across different tumor types, so you don't have to repeat the same experiment over and over again.
Yeah.
So moving on to rare diseases and unmet need populations, Elad, in your clinical experience, what are special considerations for those very rare tumor types?
Yeah, well, let me follow on. Actually, it's something that Julie just mentioned. Some years ago, we discovered really that MSI-high patients, or those individuals that have mismatch repair deficiency, are more responsive to immunotherapy. And so that became actually a rare subset of a more common set of tumors, several that actually have regular responsiveness to these types of issues. Actually led to the first, maybe the second, tissue agnostic approval by the FDA in the form of pembrolizumab. Over time, though, nivolumab and ipilimumab also ended up getting an approval, at least in colorectal cancer, for the combination of for those patients that had mismatch repair deficiency. What's interesting, just for me, from the real world perspective, is that patients are typically not getting that particular combination, at least not that I've seen. Maybe that's something I could look at in real world evidence. People tend to actually use the single agent, the pembrolizumab approval, in that setting. And it's maybe unclear, to be honest, even though they were able to obtain an approval, that nivolumab and ipilimumab is necessarily superior for those particular patients. It's something that, because it was never really tested, it's something that, at least as the community oncologists, have been less likely to take that extra risk of additional toxicity and subject their patients to that when they might actually be able to get a benefit from just the single agent. And that's despite the fact that we get great data from people like Miriam Chalabi, who out of the Netherlands Cancer Institute has actually been able to show that nivolumab and ipilimumab in her Niche 2 study had tremendous responsiveness for those patients with mismatch repair colon cancer. But then again, we've gotten drugs like dostarlimab approved for rectal cancer from very, very small numbers of patients who have mismatch repair deficient rectal cancer, which is very, very similar to colon cancer. It's just a different, place, right? So it's the rectum versus the colon. We tend to think of it as being very, very much on a continuum. And there they actually got 100% response rates from a small trial out of Memorial Sloan Kettering that led to the lack of a need for disfiguring surgeries, radiation, and further chemotherapy. So the contribution of effect can actually really be quite important, even if you have an effective therapy like nivolumab and ipilimumab that's been proven again and again to actually be useful to have something above and beyond the single agent. You don't always know you need it unless you're actually testing it in the right setting.
Yeah. Great points. And I guess 100% CR rate, you probably don't need a randomized study, right? Yeah.
You can do that later if you feel like it.
But the other point you bring up, and I think we discussed this a little bit in our panel, even if you get regulatory approval, if you haven't really demonstrated full contribution of components, patients, clinicians may be wary, of the added toxicity or other burdens. So
legitimately,
it doesn't mean that that it'll be utilized widely in the clinic. Okay, so final question before we turn to audience Q&A. Looking ahead, how can we make combination trials more patient-centric while generating the evidence needed to understand contribution of effect? Julie?
Yeah, so this kind of goes a little bit on what we talked at the last session, is that I do think that we need to potentially reevaluate the use of more adaptive designs and combination studies so that we're, adapting the randomization as the trial goes along to make sure that patients are being placed into the arms that have the best risk benefit. I think that, those trials are not utilized quite as much as we would like them to. For one reason or the other, they're probably highly difficult to negotiate with the different regulatory bodies, et cetera. But I do think that this is a very patient-centric approach to making sure that, arms that don't have good monotherapy efficacy, et cetera, are being dropped. Or maybe are too toxic even in the combination setting, right? It would be an early stop. And then the other one is going back to what I said a little bit earlier with the other question, is the concept of leveraging model-informed drug development throughout this journey of combination development, right? And that starts from the very first patient that's enrolled in your monotherapy dose exploration into your combination dose escalation studies all the way into phase 3, leveraging, the different exposure response analyses that we could potentially use to not only help us decide doses for combinations, but maybe apply for contribution of component exercises to reduce that full factorial design, I think is really important, and I think is something that we'll hopefully see more of in the future, in addition to the model-based meta-analysis as well.
Great, thanks. Carol?
Well, I think patient centricity for combination trials is the same as it should be for any other trial, really. And I like to think of it as start by remembering that patients are unique and they represent their own story. They have a family behind them. They have others who care and love and support them. And there's a need to work with patients to really communicate the uncertainty when they are asked to join a trial, and being very clear of what you're asking them. I think trying to reduce the burdens on patients, even things such as how many blood draws will you need? How many imaging will you need? So I live in Vermont. We're a small but rural state. And it's not unusual for people, for clinic visits, to have to drive over 100 miles just to see their provider. Now, if you're doing that once a week for 12 weeks, or I was hearing about a protocol yesterday that was an image once a week for 12 weeks, that's a lot of burden to put on patients. Also, I think patients need to be assured that they're not being unnecessarily exposed to toxicities, and gathering information on quality of life issues, including patient-reported outcomes, I think is really important to patients and to the studies. And I think it's critical to have and I go back to this a lot, but very clear informed consent documents so that patients and their caregivers and their families know what they are agreeing to and how they might be harmed or how they might benefit from this research, or how others in the future may benefit from that. So, to kind of go a full circle, and I think I read about this in one of our papers or somewhere in a slide, it's this balance between efficacy and safety, and combination should enhance outcomes without toxicity and redundancy. And I think that's the best way to put patients really at the center of the studies.
Thanks. Gary?
It's hard to build on that, because I think that says it all. I think it's a real, it's incumbent upon all sponsors, in order to reduce the burden and complexity of our clinical trials. We need to be prioritizing decentralization where we can, meet the patient where they are. We need to be reducing the number of unnecessary assessments that make their way sometimes into our protocols while balancing the scientific rigor that we must also be prioritizing. I think Carl's really made these points super clearly. And that's across all of our studies, regardless of whether contribution of effect is relevant or not. Where contribution of effect is relevant, it's making sure that we have studies that are attractive, that patients would want to enroll and that investigators want to open at their sites. We've talked about the individual contribution of effect arms that can be in these factorial studies. And we need to be thinking about the totality of that. Patient-reported outcomes, I think, has just come up from Carl. That's something that could potentially support contribution of effect as in the totality of the data package. We've got a lot to do.
A lot to work on. And Elad, any closing thoughts before we move to Q&A?
Yeah, I mean, I think that I would argue that we need to bring these innovative trials more to the community as much as possible, and that automatically, I think, addresses some of the issues that Carol and Gary actually both brought up. In the sense that if you're going to be doing a clinical trial that's being conducted in a community oncology site, you will understand the compromises that you need to make early on. Yeah. If the effect is significant enough, which one would hope it would be, then you'll see it when the drugs are actually administered in the same setting during the trials that they'll ultimately be administered at in the after the approval setting. Basically, we're talking about in the U.S. 80% of patients basically getting treated in the community oncology setting. So doing everything for the academic sites, preferencing the academic sites, even though I'm from one and I think it's important to do, is short-sighted ultimately. One has to actually think about the community and academic collaboration to actually move forward.
Great points. Okay, so our first question, and this is, I think, very timely because we're all sort of struggling with the post-IO setting and how to isolate contribution of components. So in situations where one drug, such as, for example, an anti-PD-1, PD-L1, is used in first line, and the combination for second line includes that drug, how should we consider COE? So, who wants to take a stab at that? Elad?
So I had a trial design that I love that we used towards the end of my time at the NCI, which involved essentially 2 experimental arms. Where we would compare both of these experimental arms to a historical control initially, just to get over some sort of threshold, but randomize patients to both of those experimental arms. If both arms make it past that threshold, whatever historical threshold you have a formal comparison with whatever endpoint you think is appropriate, PFS, response rate, what have you, so that you're actually at least able to prove that something is better than, let's say, the monotherapy, push essentially your drug development forward. This was something I published with Jared Foster, who's one of the statisticians at the NCI, and we published in the journal of the NCI in the past few years. And that trial design, I think, addresses some of the concerns that we have here, that first of all, we can get information about monotherapy, we can get information about how the drug actually responds when that new experimental drug responds in combination with PD-1 compared to to that drug on its own. And it also helps patients get to the right therapy faster, but with proven evidence. And we certainly used designs like that in the past. SWOG did a trial for patients that in melanoma who progressed after PD-1 monotherapy, comparing essentially nivolumab and ipilimumab to ipilimumab by itself. again, including the PD-1 therapy on its own to really show that that combination was what was driving the benefit for those patients in a randomized fashion. And unfortunately, for contribution of components, contribution of effect, that's the only way to go.
Yeah, Gideon, I think it depends on the mechanism. I mean, this is a really interesting question because, like, in most instances, if you've failed PD-1 in first line, putting it into second line is questionable. But I think it depends on the combination and whether or not they're retreating with the same PD-1. It depends on the mechanism. Like, if we're just trying to make PD-1 great again, right? I mean, so. That's a different question. Yeah, we need a hat. Yeah, make PD-1 great again. Maybe not red though. Because I've worked on a couple of different assets like this when I was consulting, and it wasn't for PD-1, but it is a very difficult space to be in because you don't want to necessarily re-challenge them with the drug alone. But you do sort of need some of that data in order to understand whether or not a PD-1 or another PD-1 could be as effective as the combination that's supposedly trying to make PD-1 great again. It's a really difficult space to be in.
The way I would just add, the way I think about this is, if I was a patient, would I want to go on to the same treatment or the same class of treatment that I had just progressed on? And the answer is, for me, unequivocal no. And there is evidence on re-challenge in certain settings, but it's not the same as a patient who has just progressed on this. And I think this is a great example of where methodological rigor is really at odds with the clinical situation and the reality that these patients find themselves in. And I think it's up to us to have studies that are actually attractive for patients to want to enroll in. I'm not sure that a monotherapy PD-1 arm after failure of PD-1 is really appropriate.
Right. Yeah. And I guess it's also discerning what's, sort of pseudo-progression versus true progression. If it's true progression, then absolutely it wouldn't be ethical to continue a patient on the PD-1. And actually this next question flows very nicely from the first question. So how does historical data in the literature weigh in when considering exclusion of monotherapy with PD-1 therapies? Can sponsors successfully reference literature as justification not to include a monotherapy arm in a trial? What are thoughts?
There was a wonderful analysis done by Brian Topp at Merck, actually, some years ago, where he looked at those patients that were taken off trial in a variety of large trials, or at least had been declared to have progressed, and then saw those individuals that continued to be treated anyway because the trials actually allowed for it. What was interesting was that the response rate in monotherapy PD-1 or monotherapy pembrolizumab from that new baseline was actually in the low 20s in some melanoma in the melanoma analysis. It was a little bit lower for others, but it sort of speaks to the point that immunotherapy was a little bit different and still is, and may actually have some responses even at unexpected time points. It's hard to do, I agree. There's a good reason why patients might not want it, but it does make it a little bit more difficult and challenging from a statistical perspective to run those clinical trials. And so our goal essentially was to show that those new agents that were being added to the PD-1 were ineffective, that justified us moving forward to a combination with, let's say, a PD-1 and the new experimental agent.
Yeah, great point. So again, the methodologic rigor is crucial. Yep. All right, moving on. Maybe I'll ask Carol to comment on this one. So how should crossover be considered in COE, and is no crossover acceptable from a patient standpoint?
Well, I hate to speak for all patients, so I'll speak for this patient or potential. I really like crossover in the design because that way you are getting both or whatever, how many other treatments there might be. So it seems like it's a lot less risk for a patient to say, yes, I'll be in this trial. So I'm not saying it's ever not acceptable, but I would really prefer to see crossover designs, speaking from a patient's perspective. So I would endorse that.
Yeah, great to hear. And there have been examples in the past where, sponsors wanted to get, OS for, demonstrate the overall survival advantage, and actually the agency, in these studies where there seems to be tipping to the point of losing equipoise, encouraging sponsors to cross patients over. But it is a tricky scenario because obviously there has been a renewed emphasis on making sure that OS, not only there's a trend, but OS is demonstrated. So it's always.
And I would assume there's always a timing issue also with where do you go first and when does that happen in terms of crossover?
Absolutely. Okay, next question. Can you speak more about biomarkers, ctDNA, cfDNA, especially if not clinically validated, but perhaps research validated?
Why are you looking at me?
You're our pharmacologist.
My pharmacologist.
Yeah, early
I'm assuming they
developer.
want us to speak about it in combination, just not just randomly about ctDNA. But so I think that the trends in ctDNA and other biomarkers, are incredibly helpful, and the totality of evidence exercise when working in combination and helping you figure out your contribution of component exercise. They can also obviously be very helpful when we're talking about dose. And not all of these things are going to be clinically validated. And I don't necessarily strive for them to always be clinically validated. I think that when we're talking about clinical validation, we're linking the ctDNA to an efficacy endpoint. Whereas, if it's not clinically validated, we use it as a tool to sort of provide us with, a looking glass as to whether or not our drug's activity is actually working in monotherapy or combination, whether combination is giving us more of a response than if we didn't. And I think that that's still very informative. We don't have to have everything to be clinically validated to still help us inform and make decisions in drug development. And so that's how I feel about, using biomarkers and dynamic endpoints and this exercise.
Yeah, and I know Janet Woodcock used to shy away. She hated the validation, V word. It was more like qualification rather than validation.
Yeah.
But I don't know, Gary, any other thoughts on ctDNA? How do you guys use it?
I completely agree. It's fantastic technology. It gets better and better all the time. It can give us very early looks to the utility of monotherapies and combinations. It can help us look into the future, as you said. We know that this is directly looking at anti-tumor efficacy. The difficulty is the validation in each setting towards clinically validated endpoints and the regulatory view on that. But I'm hoping that these conversations are going to evolve very positively over the coming years.
Great. And Elad, how are you using it in the clinic?
So they are being used, quite a lot actually in standard of care already. One thing I would say is that they're not yet clinically validated, but so much effort is actually in the direction at this point with circulating tumor DNA and cell-free DNA, and there's so much need, obviously, to get something a little bit more precise than the imaging-based criteria that we've had, that I do think that within, I'd say 5 years, there will be a lot of clinical validation in a variety of different settings. And we already saw it in the setting of uveal melanoma with an Immunocore agent that got approved, tebentafusp, very hard to say, that showed overall survival advantage to standard of care in uveal melanoma with those patients who were HLA-A2, but those individuals also had a cell-free DNA component that showed a benefit that was noted on clinical trial, even though clinical validation was not yet available.
And I would say patients are very interested in moving to that clinical validation. It's a lot of excitement in the patient community about that.
Yeah, much more patient-friendly to forego a tissue
Exactly.
biopsy.
Exactly.
Get a blood
Lots
draw.
of imaging, no radiation, all of that, yes.
Yes. All right, so we've been informed we're at the 3-minute warning. This is the last question. The panel's thoughts about efficient starting doses for novel combos. Both haven't identified MTD in prior dose escalation studies. Julie, I'm sorry, but I'll have to look at you again.
Good thing Julie's here.
It's totally fine. So my first advice is to not start at MTD for the combination. So if that's not, hopefully that's obvious. And I think, I'm kind of anti-MTD. I don't really think that we need to be identifying this and taking it in for the majority of our assets. I think for some ADCs and some cytotoxics, it makes some bit of sense to still be looking at MTDs. But I would be looking for efficient starting doses, I would actually be looking back at the data and monotherapy from a dynamic perspective and a target engagement perspective. And I'd be looking at those, where do those tipping points plateau for each of the assets, and moving from there. But I don't think you need to start at a non-efficacious dose. I think you need to start at a dose that makes sense from both an efficacy or activity perspective. And ideally, obviously, we'd be looking at the safety of these two drugs too to understand what the potential overlap from that perspective would be. But definitely don't start at the MTDs. And MTD -1 is not also typically good enough either, yeah.
Okay, and just kidding, we actually have one more question after this, but Gary, any thoughts?
I think it depends on the mechanisms of action as well. I would just add that. So if you've got, for example, an immunotherapy and a targeted agent where you're not expecting overlap in mechanism and non-overlapping toxicity, I think that can be a very different situation to some of the ones that you've described, Julie.
Yeah.
Yeah, I think we need to strike out this MTD concept, go with optimal biologic dose or otherwise from the vernacular. It's tough.
It's tough.
Yeah.
It's a habit. Yeah.
Elad, any thoughts on the dosing question?
Yeah, well, so one thing I guess I would mention just to add is that there are some drugs that actually have just extremely wide therapeutic indices. And so, we definitely saw that with PD-1 and PD-L1, where very low doses, still have an effect, and very high doses have an effect, and people have been playing around with that for a little while, but without much cost, actually. Whereas with some drugs like CTLA-4 based, so it's not different just because it's immuno-oncology. CTLA-4 is obviously an immuno-oncology agent, but there's definitely a dose response and a dose toxicity relationship. So I think it does vary, considerably, but biologically, the biological active dose or the biologically most efficacious dose is obviously something that we ideally should be figuring out in those early stage trials. You can't do it if you don't test it.
Yep.
Yep. And Carol?
I disagree with, I agree completely. I just agree completely with the new terms as opposed to MTD. They just sound much more patient-friendly and not like you're trying to see how much can you manage, how much can you tolerate before bad things happen. So, yeah.
Absolutely. Okay, and the last question, and it's a good, we learned a lot from the morning session on seamless adaptive design, so I think the answer will be yes, but I'll await the panel's responses.
Yeah.
Could adaptive studies be used to drop arms for COC during a study or an early futility style analysis? Gary?
Yes.
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 All right, there you have it. We have consensus. All right, well, I'd like to thank the panel for an engaging discussion, and I think we have lunch coming up, but turn it back over to you, Jeff.
Thank you, guys. Really a great, lively discussion and very thoughtful. As Gideon said, we will now be breaking for lunch. You should be able to find multiple different food stations out in the foyer behind you. I would invite you to grab lunch, take some time to network with your coworkers. You're welcome to bring lunch back into this room. In about 40 minutes, we will reconvene with a panel of leading experts who will be providing their perspectives on what we can expect from policymakers in the weeks and months ahead. And I will warn you, they have all the answers. So we will see you at 1:20 to get started. I think they cut that off. All right, come on, where's Roy?
If everyone could please be seated, we're going to welcome our lunch keynote panel to the stage.
You are third. You're first. You're on the end. And I'm not sure, there's Roy. Yeah, you're on the end. Yeah. All right, thank you. We will go ahead and get started with our next session as everyone grabs their seats. I hope you had the opportunity to, of course, grab lunch, but also connect with colleagues during the break. During the course of the event, I hope that you have had the chance to note several of our upcoming events scrolling on the screens. I want to highlight one more that will be coming up, a virtual event, an advocates webinar that will take place on November 17th to continue the discussion around the important sessions that have been the point of discussion today. And you can find more information about that on our website to register for the Advocates event, as well as any of these additional events, or scan the QR codes on the screens. As we make our way back from lunch today, I'm very pleased to have four remarkable individuals here to help provide their insights onto current and future policy. I was joking, a couple of you, if you weren't here before, I promised that all the answers would be disclosed during this session, so we're very much looking forward to your insights and what you have to tell us about what's been going on in the world. But I will skip formal introductions. Most of these folks, I think, need no introductions, but you can see the information about their bios in the meeting material by scanning the QR codes and what's available at your seats. So let's just jump right in, okay? Maybe, Remy, I'll start with you if that's okay. It's kind of the million-dollar question, I suppose, as we're now entering the longest shutdown in government history. Any thoughts on the timetable for Congress to make a deal at the end of the structure, to end the shutdown?
Yeah. Well, it is the million-dollar question, and Jeff and Friends of Cancer, thank you for having me back. I appreciate being here and always appreciate being in front of this group and being a part of this room. Government shutdown, yes, we're in the longest shutdown. If you haven't been paying attention, FDA obviously has a lot of more resources than some of the other agencies given the user fees and the carryover balance there. But we are expecting some logjam to break this week. We may have a vote on the Affordable Care Act enhanced advanced premium tax credits to let some air out of the balloon for both sides and get these folks to end up back at the table. The conversation as of last night was potentially doing some sort of combination, continuing resolution for things like the labor H budget, potentially a minibus with things like the FDA ag budget, and then being able to sort of punt a lot of things potentially into January. Some people are saying December. Speaker Johnson doesn't want December. And then some folks are saying, let's just clear the decks. Let's get it all done in one big vote and try to get through September 30th of the next year. So it is still very in flux day to day. But we are hoping that we can start to move forward and have some of these conversations beyond just the government funding piece of this. I know there's a lot of riders folks care about, general health care extenders, and then policies like the pediatric PRV program that need to be reauthorized that did not happen at the end of last year. So a lot is riding on what happens with the government funding package.
Does one side come out ahead?
I think they both think they come out ahead right now. Obviously, we have an election today. If you haven't voted, go vote. And we'll see. I think Dems feel like they are winning the messaging on healthcare. I think Republicans feel like they're winning the messaging on, hey, we tried to keep the government open. Democrats didn't want to do that, and that's why we're seeing some of these detrimental effects on SNAP and other programs. So I think we need to get sort of through the shutdown and do some retrospective before we know who sort of won this round.
Okay. Roy, things over the last several months have been, in the health policy world, somewhat unsettled. For your institution at Yale, how have you seen the impact of sort of this unpredictable environment at times?
Well, we've been tremendously concerned. Yale has gets some of the highest amounts of money from the government, and when we hear there are going to be 40% cuts, in the NIH and NCI budget, that's extremely worrisome. Now, we were happy to hear that Congress was looking to bring that back, but we need Congress to come back into session to do that. But then, of course, there are the effects on the indirect costs. Large academic institutions like myself, we have indirect costs that are in the 60, 65, 70% range, and a cut of the indirect costs to 15%, that would have a huge change as well. If that's not enough, I work at a hospital system, cuts in Medicaid, especially when you're a safety net hospital, could be huge. So that's quite big. And then to top it all off, we also have the threat to the endowment. And Yale's in the highest group, and I guess we should feel fortunate for that, but certainly an 8% tax on the interest on the endowment starting in January is concerning as well. All this as we try to get young people to go into science, to get their first R1s. The good thing is, I will tell you that the NCI paid out many of its grants in full this year, but the question is what's going to happen next year and the year after. And we're hearing, funding rates at the 4th percentile, which is it's going to be really, really tough, especially for young investigators. So we're working hard to sort of provide bridge funding. Some grants have been cut because of policy. Grants that work on underserved populations and health disparities, some of the global work that we've been doing, which I think is so important. But we're trying to budget for that, so we're being more frugal. Initially, there was a pause in spending. Some things as simple as you don't get lunch anymore at Grand Rounds, so no one comes to Grand Rounds. But those are trivial things. But I think more important, now I think we are going back to very selectively rehiring, and the work needs to be done. And I think the institution, will go on, but it's going to be a challenge. We're going to have to look for other ways to fund our initiatives. I see so many industry leaders here today. Public-private partnerships, something we've talked a lot about at these meetings, that I think are going to be even more important. The clinical trials need to go on. Patients need to be seen. So it's a time for caution. We're hoping that when Congress gets back, they'll at least give us a flat budget so that we at least, it's not so dire. And then we'll just have to sort of, I think we will, of course, be more creative and try to innovative, but it's a time that we have to be worried about.
Yeah. Anna, despite all of this uncertainty, FDA has rolled out several new initiatives. They're keeping the policymaking apparatus going and trying to examine various aspects of efficiencies, things like the commissioner's priority review, pre-check for manufacturing. The use of AI has been top of people's minds. How significant do you see some of these exploratory programs rolling out over the next several months?
Yeah, well, Jeff, thanks for the question, and also delighted to be back with Friends of Cancer Research here today to talk about issues that are so important. So the short answer to your question is time will tell. I was reflecting, as today is election day, that it was just a year ago that now President Trump was elected to serve a second term, and I think fair to say that his team didn't just hit the ground running immediately after that. They hit the ground sprinting and have not looked back. And so when you look at the cadence and the ways in which FDA is leaning in on priority issues, really coming from the President, the White House, HHS, in furtherance of their mission and how they can be part of that, we're seeing it's being met with a similar type of speed of hitting the ground and trying to move quickly to demonstrate progress against that. I was also reflecting today that it hasn't even been yet, if my math is correct, 6 months since the Commissioner's National Priority Voucher Program was announced. They've been rolling out additional details. Just recently, the agency announced the first 9 recipients of the voucher. In rolling that out in that announcement, they indicated, and dare I say previewed, likely more are coming. So I think that sets an expectation that we're going to continue to see the agency looking at within their mission where they can lean in on making demonstrable progress against priorities that are consistent with what the administration is seeking to achieve. And much of that is really rooted in an America First initiative. So you mentioned PreCheck, right? That's part of strengthening America's domestic manufacturing footprint, thinking about how to streamline, cut down on regulatory steps that can be modernized to be more efficient in its application. And certainly leaning in on innovation was a theme that was part of the first 4 years with the first Trump administration and continues to be one in this second term. And so looking really to try to ensure the agency is as modern and efficient as possible in executing its mission. That is not a new question. Commissioners and agencies is a question we're constantly asking, right? It's a question Congress is constantly asking as well. But at the same time, I think they're somewhat answering it in the moment, and will continuing to be seeking input and leaning in where they think that they can make progress against those objectives.
Yeah. And it seems like the process, at least in terms of the priority review candidates, each could be a little bit unique given the diversity of products that have been nominated to that program. But all of these changes, Amy, oftentimes can bring additional adversity from a staffing perspective at FDA, and FDA staffing has always been something that needs to be well supported. And I'm curious what your thoughts are around how the current environment at FDA is affecting, at a review staff standpoint, just giving your past experience of having spent several years there.
Yeah, so we're heading into user fee negotiation season, and we haven't gotten any indication from this administration that they would not support continuing to support new hires at the agency to conduct reviews appropriately. It's always hard to recruit at FDA, even without the challenges of the current environment. I mean, no PhD or PharmD or MD student goes to grad school thinking, I'm going to work for the FDA. It's just not a career path that's obvious to a lot of people. And so I think the challenge of the current environment is when you hear about the shutdown, all the details of shutdown don't filter out to potential hires, right? They don't hear about the details of the carryover funds, and they don't understand that, the review staff is still working right now and is still getting paid with these carryover funds. But that in itself is challenging as well. I will say from the AZ perspective, we have not seen any balls dropped in terms of our own submissions. So they are working very hard. I do think it's an incredible career, and I enjoyed all of my time at the FDA. And so anyone who's thinking of going to it, I would highly encourage it, because I think it's a great place to do a lot of learning. But it's challenging in this environment, and especially during a shutdown. Yeah.
Oh, it's heartening to hear the positive thing and the continued interactions. Maybe I'll stick with the positive side of things, Roy. I think you recently just came back from one of the leading oncology meetings each year at ESMO. Any thoughts on despite some of these challenges, where's the science heading? What types of results were presented at that meeting and others recently?
Yeah, so ESMO was in Berlin, and it was just extraordinary, the amount of science. So that's the good news. And I think, basic principles, we're taking our best drugs and we're moving them earlier in disease. We're finding patients earlier with screening, and we're seeing great effects, like in breast cancer, bringing the HER2 ADC in HER2, the DESTINY trial, bringing that either to the neoadjuvant or the adjuvant setting, changing practice, and taking a disease and really making more and more patients cured. So I think things like that are exciting. Also, immunotherapy, immunotherapy being added to antibody drug conjugates. I was struck, there must be about 500-600 of these now, in study, but several of them, really very positive results. There was a keynote study adding pembrolizumab to Fortin and Vendotin, and with very, very nice results. So things are really moving forward. I will say that, I was struck that a number of the studies were from China only. I think that's something I think from a policy point of view we have to start to think about. There were 2 lung studies that were exclusively done in China. One was a PD-1 VEGF combo, and this is all the rage right now in the lung cancer world, and that's my area actually, lung cancer, still the number one cause of cancer death, and a trial that was completely done in China. And then there was a second trial of an ADC, a TROP2 ADC, also completely done in China. That one actually had a positive survival result published in the New England Journal of Medicine. So I think what we're seeing is a great deal of innovation, ADCs, bispecific T-cell engagers. We're seeing combinations. We had the sessions today. The goal now is to get these drugs to patients. And one of the reasons I was so excited to get the invitation to come today is that's the goal of the Friends, to get drugs to patients as quickly as possible and to get through these regulatory hurdles, these other issues. And I think that we have a great deal of potential.
That's encouraging to hear. Hopefully the science will continue to lead the way. Amy mentioned, actually Roy mentioned some positive results that the AZ was part of recently coming out here. But as you think about some of the environmental factors, I suppose, from a policy standpoint that are creating a downward pressure on industry, how significant are they, and are they having an impact on how a large sort of global organization like AstraZeneca are thinking about the future of R&D?
So I would say we are as committed at AZ to R&D as we ever have been, and I'll lead with 2 stats around that. CDC recently, a few months ago, released statistics in the US around causes of death, and cancer has regained number 2 after the COVID blip. And I think everyone's familiar with all the reports lately about cancer diagnoses increasing under the age of 50. So we clearly have a lot of work to do. And I think that there's unmet need everywhere. I think we're trying to develop therapies, especially for those younger, not especially for, but also for those younger patients that have more effectiveness and not too much toxicity because they are expected to have decades of life and we want them to have that. So there's a lot of unmet need still. And AZ, thankfully, we are led by science and we are able to invest in all of the therapies that Roy just mentioned. We invested in radioconjugates, we've invested in cell and gene therapy. ADCs are our sweet spot right now. We have small molecules, we have T-cell engagers, we work in both the solid tumor and heme space. So we are poised to continue to invest as much in R&D as we have in the past, and we're not going to stop because we still see that unmet need.
I guess maybe for either one of you or both of you, given the promise of these agents and sort of all of the different mechanisms that are being explored here, is this unique? Are we at a unique point in time in scientific history with pipelines that are apparently so deep?
I think we're at a unique point in history that we have so many tools to help us develop drugs. We use AI in end-to-end development at AstraZeneca, from computational pathology and biology to help inform how to make the molecules, foundational models to help select the right patients to make sure that our studies are successful. So I think we're at the cusp of another point of acceleration. I would say that when we got the immuno-oncology agents to clinic, that was from decades of research that finally got us to clinic, and that was an acceleration point. And I think we're right on the edge of another acceleration point.
Yeah, I couldn't agree more. I think back to the Hanahan and Weinberg Hallmarks of Cancer paper, 2000, in the year 2000, and all the different pathways that are thought to be involved. We have those tools now. We have the drugs, but we're not done yet. We have to find the right patients, the right biomarkers, faster trial designs. But one company might have the whole pipeline, but I also think that it's going to be up to us in the academic world bring all the companies together, because we're going to have to. It's going to be combinations, as we've heard about today, and it's going to have to be the right combination because sensitivity, resistance are still the major issues. But yeah, I think now's the time. We're accelerating. We've got to make it happen.
Amy mentioned earlier that we are, this week, I think, formally maybe moving into user fee negotiation season with the start of those discussions occurring. And yes, it's a long process, but one that is sort of a milestone moment in FDA policy each time it finds itself coming around. What do you see as some of the topics that will likely be discussed as part of this reauthorization process?
Yeah, so I'll start by noting that it feels like we'll focus here on PDUFA because it was the original, it was the OG of the UFAs and kind of set the stage for those that followed. It is always a reflection of the moment in which you get around a table, first with the negotiation between industry and the agency, and then when the commitment and the recommendations are sent to Congress, and then the work that happens with the House and the Senate and Energy and Commerce and Help coming together to think about, okay, what might need to be added on to round it out, right, from a policy conversation. So I think just bridging from what Amy and Roy talked about, this will also be a moment, right, of reflection of where is there an opportunity, where does industry, patients, everybody who's part of this ecosystem looking to see more certainty on the path towards really bringing forward these innovations, because as exciting as that is, a core tenet of PDUFA is that the agency has to be prepared for the innovations that are coming before them. There needs to be clarity, not predetermining the outcome, the regulatory decision, but there needs to be clarity on the path that it takes to get from point A to point B. So when we have a backdrop of uncertainty, in the midst of that, I think very much part of this conversation is going to be where are there opportunities to be creating greater regulatory certainty for the innovations that we're seeing now and those that we're anticipating to come within the next 5-year cycle of that reauthorization. I think if you look at some of the comments that have been made by administration officials, there's been references to simplifying user fees. We'll see what that looks like. Again, time will tell, right, as that plays out. I also think workforce is going to factor into this, and I'll just take a moment here to reflect. We're seeing quite a bit of attrition at the agency. A couple weeks ago, FDA posted as part of their PDUFA commitments, they do certain reporting requirements around hiring targets. And recruitment and retention has been one of those perennial issues that every UFA, there is attention on this. Congress in a bipartisan fashion has worked together to try to put more tools in FDA's toolkit to really recruit and retain the top-notch talent, because being prepared, there's a resource alignment, but people are the resource, right? That they are able to understand and work with those things coming before them. So I would anticipate workforce is going to be one, given what some of the latest data coming out of the agency shows in terms of keeping pace with where they need to be for the anticipated workload. I would also make an observation. It'll be interesting to see in previous PDUFA negotiations, you've seen a commitment to issue guidance on topics. Guidance is a great tool that the agency has to help to share the agency's, it's not binding, but current and best thinking on a topic, and you can also solicit comment, and it's a very iterative process to helping the agency keep pace with innovation and science coming before it, and an opportunity for input from stakeholders, of course. And so when you're in a 10-to-1 deregulatory paradigm, right, I think it does kind of cast an interesting question of, are we going to see as many commitments of leaning in on guidance as a way to be demonstrating progress and working through issues? And if not guidance, what then to speak to that? Or do we see the contours of what comes out of this next negotiation maybe different from what we've seen? Because again, it's going to be a reflection of this point in time and anticipating what's coming further. Lastly, I would just say, rare disease, Remy touched upon the pediatric rare priority review vouchers. I think rare disease is increasingly always an area, right? And I think to the agency's credit, even in the midst of some of the uncertainty and changes that they've been navigating, they're continuing to make progress against PDUFA 7 commitments, right? And continuing to foster a patient-focused drug development paradigm. So I would expect, if we've seen with others, we'll continue to see the next kind of iteration of what that looks like that is reflective of where we are now and where both the agency, industry, patient groups, kind of the whole ecosystem that then participate in this process, see it going forward.
That's very helpful. You guys have both had, front row seats from different sides of the stadium, even, I suppose, in different reauthorizations. But, as you've thought back on sort of where we are with the kind of political moment and gridlock that is currently being observed on the Hill, Remy, do you see this particular reauthorization cycle potentially being more challenging than in the past because of that?
Yeah, potentially. And I always like to say that, it is going to be 2 years from now. And some of these last few months have felt a little bit like dog years. So I don't want to sort of predict what's going to happen. We're going to have a brand new Congress at that time, right? In theory, these letters will, sort of the negotiations wrap up in the spring. They get sent to Congress officially in January, but those briefings will happen at some time next summer. But I agree with Anna on the, these agreements and then the legislative package is always very reflective of the moment. And a lot of the priorities of the administration have been bipartisan priorities and are largely generally bipartisan ideas, whether that's, as Anna mentioned, things like workforce, even things like NAMs or new alternative methods, right, to reduce animal testing. That's always been a bipartisan issue. A lot of ink was spilled on that issue within FDA under the Biden administration and more under this administration. Things like how do we get more folks to onshore here? Are there different incentives that we can use? Are there things that we can do to be more competitive, especially in sort of the launch and clinical trials space, to keep on pace with what we're seeing in some other nations who are leaning in maybe in a bit of a different way than FDA? And so are there ways that Congress can push forward there? And so I think that there are a lot of bipartisan places, and I think there's places where, depending on how some of FDA's current initiatives go, there may be an effort to maybe codify those, right? You have something like the Commissioner's National Priority Review Program. Maybe folks turn out to really like that. Maybe that works in a certain subset. Maybe, we like that for, Jeff mentioned that there was a lot of different types of drugs within those 9 that were initially announced. Is it appropriate in one case or another? Does Congress want to step in and codify that? Potentially. Do they want to step in and codify something like PreCheck to make it easier to have new facilities here? And so depending on how some of those pilots go, I think there could be an impetus to try to codify things that are working and give it that regulatory certainty, give it that legal certainty, and not be in sort of a perpetual pilot phase with some of these things.
Roy, sort of keeping on this theme of future opportunities, I suppose. The panels today have focused in large part on current challenges in clinical development. When you think about new ways of clinical investigations, where do you see future opportunities around the conduct of clinical trials? You've been a thought leader in this space, overall for our organization as well. But where do you sort of see the future opportunities to improve efficiency?
Well, we talked about how many wonderful compounds and targets there are, but they can't all go to every patient, and everyone can't go to phase 3, and we need smarter trials. So I'd say there are 2 groups. First, and actually I sat on this stage, was it about 14 years ago?
Probably close.
Last time I had any free time from Ellen, because we started the lung map trial. So the idea was to match the right patient to the right drug with biomarkers. Now, those trials were designed to test drugs across the country, so access, diversity, and then find leads that could then go forward. So I think we need more trials like that with biomarkers. I didn't mention it, but at ESMO, I was struck by ctDNA being used to tailor when to stop therapy. I don't think it's ready for prime time yet, but smarter trials with biomarkers, find the right targets and the right combinations earlier. But then getting the trials done faster and in more rural and diverse populations, under-supported populations. Because as we know, 90+% of patients can come to Harvard, Yale, George Washington. They've got to be treated where they live. So that's where we get into the more pragmatic trials. And I'll just say a quick word on that. The pragmatic trial design actually came out of the lung map trial. And it really has a lot of history here at the Friends meeting, because we did a lung map trial where we took 2 drugs, pembrolizumab, a PD-1 inhibitor, and ramucirumab, a VEGFR2 inhibitor, and we combined them together. And we saw a signal in the lung map trial. So then the question was what to do next. We got breakthrough designation, something that we heard about at the beginning of this meeting. But to do a $20, $30 million trial that would take 2 or 3 years, no one was interested in that. The companies weren't, the investigators weren't. The FDA, said, what about doing a more pragmatic trial? So we did this pragmatic trial, Lung Pragmatica, which basically we took all the bells and whistles off the trial, and the only endpoint was survival. We had 2 drugs that were already approved for different indications, comparing them to standard of care, and we got that trial out to 800-900 patients in less than a year. Now, the result didn't result in an approval because the trial was pretty much equivalent, which perhaps is a good thing, 2 drugs that are non-chemotherapy versus chemotherapy, but it set a precedent because rurality, this got to the middle of the country, this got to the farmlands. This is what we need more of. So I would say smarter trials, find the right drugs at the right time, and then get them out to the population in more pragmatic trials. And these are things that we all have to drive because it's going to involve collaboration and teamwork.
That's two great opportunities to think about how they can lead into the future. Maybe Amy, sort of sticking on the clinical trials opportunities, our last session coming up here in a couple of minutes is going to be focused on global clinical trials and how they've become increasingly important to identify and finding those patients for clinical trials that need a larger catchment. Yeah, that comes with its own set of challenges around implementing those trials and conducting them just generally. When you see an environment here in the US where clinical trial enrollment rates have remained pretty steady, at least in the adult population for quite some time, how do you think about, in the context of global development, the need to have appropriate US patient representativeness into these trials while trying to accelerate timelines?
Yeah, that is always a point of tension. You want to get promising new therapy to patients as quickly as possible, and so you would love to be able to enroll in the fastest enrolling places, but you have to have a patient population that's representative for all countries because, let's be honest, I don't want to steal the thunder from the last panel, but all of the pharma representatives on that panel are like, yeah, I'm going to do one global trial. I'm not in this particular indication. I'm not going to do 2 or 3. And so you have to try to get the representation you need for all of the health authorities represented. And the US is our gold standard health authority. You're right, we haven't nudged the numbers in the US clinical trial participation. It's a very complicated issue. Healthcare delivery in the US is very complicated, and that contributes to it. But talking about being on the cusps, I think one of the other cusps we're on is how to move data from a site of treatment to the health authorities. And it's complicated in regulatory because there are a lot of, patient privacy confidentiality issues. There's, the company who's sponsoring the study issues, and then there's the health authority issues, and you have to get them to all match up. But I do think we are on the edge of figuring out how to move data rather than having to do all of these additional things that we do. And it could cut down a lot of the expense. It could cut down on how far a patient has to travel. If we could do it through electronic medical records rather than collecting into a third site, if we can use cloud-based submissions to get the data, upload the data into one place and then health authorities can download it. Like, we're talking about all of these things within industry and with the health authorities, and we're almost there. We're so close. So talking about the next revolution, that is the next revolution of treating patients at their primary oncologist site and getting that data eventually to a health authority. We're gonna get there. We still have some things to work out, but we're going to get there. And I think that's the next revolution in clinical trial development from a sponsor perspective.
So, global development has also kind of renewed conversations around global competitiveness, not necessarily a new topic in itself, but as you think about, even some of the things that have been discussed today, or, more generally around how the U.S. is positioned currently from a biomedical research standpoint, Anna, do you see the landscape shifting, based on some of the challenges that have been described here today, or is the U.S. still in a position to continue to lead?
I think the U.S. is still in a position to lead, but I think that we are also in a position not to take for granted the dynamics that have fostered the environment for the U.S. to be a global leader, right? And Amy touched upon this, FDA being seen as like the gold standard for the world, and all of the component pieces that go into an ecosystem when you think about research, discovery, to development, to commercialization, to IP. I mean, it's all of these elements that go through and can be touched by and impacted by any number of policy changes that, one will have an impact, many will have a greater outsized impact. And I actually wrote this down to make sure I get the title of it correct. In the past year, the National Security Commission on Emerging Biotechnology came out. It's a mouthful. It came out with a report. There are a number of recommendations in that, but I think it really touched on this key question that you're asking around, are we in a position, the United States, to continue to lead as we look out in the future, particularly with competitiveness issues with China? You touched upon the clinical trial work being done, and there's a number of recommendations within that report, but I would just highlight 2, because I think that there's a synergy here with much of what we've been discussing. And there were 2 specific to FDA. One, creating a simple pathway to market and then the second being prepared for novel products. So you can also see how there's so much connectivity around these issues. I would also note a lot of bipartisan interest in this, right? And that bipartisan interest also, the FDA focus has been bipartisan because people in Congress, both sides of the aisle, spanning decades, they care a lot about these issues. These are their constituents. These are their loved ones. Everyone is touched by this. So it's also an incredible opportunity to think about how do we do it best that's responsive to the competitiveness piece, but also to align with the key objectives about making sure that a patient can receive optimal treatment in an optimal care setting when and where they need it, right? And so I see a lot of synergy with that, but I would also say we can't take for granted how we got here and then where we'll be, 10, 20 years from now.
Yeah. Yeah, I think that commitment was evident, from the bipartisan nature of this issue from, even a hearing a week or so ago that the Health Committee, had to sort of focus on these issues. And maybe to round things out, Remy, I'd love to get your thoughts on, aspects of that hearing, or even thinking about, in 2026, what are some of those issues that are likely to focus on some of these issues around global competitiveness, improved development. Are there things that you foresee the Hill particularly focusing on in the coming year? And maybe just to add to that, where would you recommend the biomedical research community kind of focus the efforts?
Yeah, I very much agree with Anna. You can tell we spent a lot of time around many tables over the years. But I think it goes back to what she said, is that everyone agrees on the problem or the issue. And that is not necessarily the case in so many policymaking discussions these days, right? Folks are coming from such different perspectives, or they have different goals. I think folks are coming to a lot of these FDA issues saying, we do want America to be competitive. How do we do that? We could disagree on how to do that, but we agree on the goal. We agree less animals in research. We agree combination therapies are sort of the future here. How we get there, they can parse through that, but being able to have that bipartisan understanding and that bipartisan commitment in so many of these areas, I think is the first step towards having a smoother process. I do think there is likely going to be a lack of movement or vehicles in 2026. There is often a lack of FDA legislating in the year leading up to the user fees, the Cures 2016 year being the exception. And I think a lot of the things that we're going to see over 2026 are really sort of priming the pump for what we're going to see in a legislative package in 2027. I don't think we've really talked about the N of 1 conversation yet up here. There is so much going on in that space, in the research community, in the academic medical institutions, even things like customized cell therapies for very ultra-rare cancers in the pediatric community. And so as we're thinking about how do we do sort of best for patients globally, folks are also going to be thinking about how do we do best for the individual patients, right, who might be in need of an individualized therapy. We have the technology to do it. How do we actually get the regulatory pathways to start to come together? There's also going to be a conversation, I think, on the sort of access and reimbursement side of the house. I know this is a very FDA-focused panel, but I think we'd be remiss unless we sort of spoke to that as well. There's a lot of pressures on that side under this administration. I think there's going to continue to be pressures under that side. And with some of the statements of the current commissioner, where they do start to talk about price competitiveness in the pre-commercial space, I think it's going to be an ongoing conversation for Congress on whether we want to continue to push into that space or whether we don't. I think for most of us in this room, it's been a bit of a third rail for many, many years to talk about costs when we're thinking about moving a drug through FDA. This administration has changed that tune a little bit, especially with the voucher program and some of the announcements that have come out of the White House. And so I do think there is going to be likely some oversight and some conversation there over the next couple of years.
Jeff, can I jump in here, which is very dangerous because the clock has run down, so yes. Ellen is going to give me an earful about this. But I just wanted to take like 20 seconds here on this point, to Remy's point. If you look back at some of the paradigm shifts that have really helped to result in positive outcomes for patients, I'm thinking breakthrough therapy here, right? It was because you had folks thinking about these issues that came to the Hill and said, we have ideas and we want to work with you on how to do this better. So just as we have a room full of people who are dedicated towards the patient and advancing the paradigm, like do not take for granted like the importance of telling the story of the work that you are doing and how it fits in with this and where you see opportunities to enhance that conversation on behalf of patients, because I think that as we look back, it's been through those dialogues that have resulted in things that have had a tangible impact in improving people's lives.
Well, thank you. I want to thank all four of you for joining us today, for your insights and sharing your expertise on a complicated time and process that will be moving forward. So please join me in thanking our lunchtime panel today. Yeah, absolutely. Well, we are going to engineer a quick switch, I think, in terms of our next panel here. We have our final panel of the day, which will be exploring topics related to control arm selection for multi-regional clinical trials. In order to move forward here, I would like to, welcome Dr. Harpreet Singh, the Chief Medical Officer at Precision for Medicine, to the stage. Please join me in welcoming all of the panel and for Harpreet's introductory presentation. Thank you.
Thank you, Jeff, for the introduction. Thank you to friends. Thank you to Mark Stewart for herding this group. We're a very lively bunch, and I hope that will come out now. Always thank you to Ellen. I'm fortunate to be a member of the working group on multi-regional clinical trials. This is a complex topic and one of tremendous importance, and I look forward to our discussion today. Sorry. One moment. Okay. Let me start by setting the stage and articulating the challenge before us. In today's environment, clinical trials must be run across multiple regions in order to enroll enough patients and to ensure a broad understanding of an asset's safety and efficacy. But we are operating in a world where there are different standards. Countries have different requirements and different levels of access. Not all regions have the same access to standard of care therapies needed for equivalent comparator arms. In some instances, the standard of care differs across regions, and often approvals for novel therapies lag behind the US. This creates a key question. How do we design a clinical trial and select a control arm that regulatory agencies will accept when the standard of care may vary from region to region? And further, how do we plan for the fact that standards of care may be at risk of changing over the course of an ongoing trial? The agency is mandated to approve therapies that demonstrate safety and efficacy that are applicable to US populations. This is something we heard very frequently from our FDA colleagues on the working group. And that these agents have been evaluated against the current standard of care. This introduces real dilemmas for sponsors. First, the high evidentiary burden can discourage global or multi-regional trials. Evolving standards of care complicate comparator selection and interpretation. And finally, some cases fall into regulatory gray zones that are not fully addressed by guidance. So where does this leave us? In the most extreme cases, some sponsors may choose not to pursue studies at all, even if it means a promising asset doesn't get developed or approved. Of course, this is not a patient-centric outcome, nor is it where we want to land. This takes us back to the topic of today's discussion. What defines standard of care in a global context, and when should control arms be reconsidered or adapted? There are two core challenges in selecting a control arm. First, when there are multiple standard of care options available. So sponsors need to decide which to prioritize without appearing outdated or non-representative. In some instances, companies select control arms with investigator's choice or that are regionally determined. This does increase feasibility and it adds statistical and interpretive complexity. Second, and less commonly, the landscape evolves. A new therapy is approved in the midst of an ongoing trial that's so monumental and transformative that it becomes impractical to keep the study as it had been originally designed. This makes sense intellectually. We remember one of the pembrolizumab lung approvals that delivered such an overall survival benefit relative to platinum-based chemotherapy that using the old standard of care became clinically inappropriate essentially overnight. At the same time, changing the control arm mid-study is incredibly burdensome to sponsors. This can impact not only the interpretability of the study and enrollment trends, but can be very operationally challenging. This introduces tremendous pressure on sponsors. They need to forecast what trials will read out and when, estimate how those study outcomes will be viewed by regulators, and assess to what extent these outcomes will truly move the needle. Together, these questions increase uncertainty during trial planning, which in turn heightens risk. These questions compound, and as the degree of uncertainty grows, companies grow less confident in their plans. This can lead to conversations around whether to move the asset forward at all. Of course, this is not the outcome we want for any of our patients, yet this is happening. Here you can see some of the strategies used today to try to work around challenges inherent to an inconsistent or changing standard of care. While each has worked in certain instances, each also has trade-offs. And we discuss the path forward in today's panel. We'll get into some of these in further detail. What's needed, though, is apparent. A path forward that's clear and consistent across indications and patient populations, a guided process for how we select comparator arms, and a widespread understanding of plausible options that can be considered. While no single solution will fit all multi-regional clinical trials, sponsors and the patients they are trying to cure deserve predictable pathways and consistent expectations. Before we start our panel discussion, I will leave you with questions our working group highlighted for the field. What approaches can support transparent and justifiable comparator arm selection? What expectations should guide sponsors and regulators when standards of care shift mid-trial? And what methods and data sources are acceptable to contextualize results when the standard of care evolves? Thank you. Looking forward to our panel. I'm going to ask our panelists to introduce themselves. I'll start to my left with Judd.
Perfect. Thanks so much, Harpreet. My name is Judd Englert, and I co-lead the late development oncology at Amgen, overseeing genitourinary cancers, heme malignancies, and supportive care.
Hi, my name is Sumitra Mandraker. I'm a professor of biostatistics and oncology at the Mayo Clinic.
Hi, I'm Kristen McJunkins. I'm an advocate with Friends of Cancer Research and a stage 3 melanoma survivor.
Hi, my name is Ray Osaro-Gammo. I'm a thoracic medical oncologist, chief scientist at the Baptist Memorial Healthcare Corporation, and principal investigator of what we used to call minority underserved NCORE. I guess we can't use that term anymore.
Hi, my name is Kathy Winson. I am a portfolio strategy lead at Genentech Roche, and I oversee the lung and pan-tumor indications.
Thank you. Judd, let's start with you. We've set the stage. Do you have any examples in your current line of work that illustrate how complex this topic is, selecting a control arm for multi-regional trials?
Yeah, thanks so much, Harpreet. So I think you've heard a bit from the panels today, and even the last one, about how rapidly the treatment landscape is evolving for oncology. And that's a really, really exciting time and exciting to be a part of. I think from the drug developer side of things, that also poses some significant challenges for us as we think about how we design trials and making sure that they're relevant and have equipoise as we move forward. So you heard a bit about how we have, this increasing, depth of targets that were now accessible, and also the breadth of modalities that are coming in all of these trials. And as we think about how they are incorporated into the standard of care for treatment, that's also obviously highly relevant for us as drug developers on how we think about how we need to design the trials so that they can be interpretable to the clinicians so that they can apply them to patients. In a disease like prostate cancer, which we're currently developing at Amgen, one of the challenges is more and more standards of care become available, where they're utilized around the world and where they're approved, as well as how they're sequenced in the variety of diseases can vary from region to region. But as you also heard, as a sponsor, we want to be able to run a single phase 3 trial for one of those indications and make it relevant to as many regions as possible around the world and to those patients in those regions as well. So as we think about that, the individual treatments are going to be fragmented in what each patient is receiving, and a lot of that falls on the clinician to kind of evaluate the benefit-risk kind of profile with the individual patient as they're thinking about that. And we need to make sure that our trials that we're designing are allowing that freedom for both the patient as well as the clinician in order to participate in something that they would be willing to be on the control arm for.
Thanks, and happy Movember.
Oh, thank you. Yes.
Kathy, you and I have had some shared experiences around this. Are you willing to share with our friends, putting my ex-FDA hat on? We dealt with this.
Yes, we have an interesting example at Genentech Roche, and this really goes to the question that we had, and we've addressed this in the paper, but also quite a bit of discussion amongst the working group about how do you define standard of care? How do you know when the standard of care has actually changed? And we've had a lot of discussion, and we listed out in the paper a number of factors that inform that, but really kind of landed on we don't always exactly know when the standard of care shifted, especially when we're talking about a wholesale shift in standard of care. So what are some of the key indicators? And we had an experience where developing a drug in the KRAS G12C space a few years back, and we were in the early stages of development, phase 1b/2, and we had a plan. And we had a plan because we could see the development landscape. We could see there were other companies developing in the same space, but we had a sense of where they were in their development, and we thought we had a pretty robust plan to implement our, the original plan was a phase 2/3 adaptive design, but as probably most everybody in this room knows, the emergence of adagrasib and sotorasib, accelerated approvals, they came into play. But these were accelerated approvals. This was early data. These were small data sets, and we were, we obviously evaluated that, and we still felt fairly confident that these are very early approvals. They still have to run the phase 3 trials. We have a few years. This won't have any major impact on our plans. And we were at a breakthrough designation call with Harpreet, and Harpreet at the end of the call said, hey, by the way, how is that trial enrolling?
Sounds like me. Sounds like a question.
And it was, yeah, she was just like, out of curiosity, how's that trial enrolling? We're like, funny you should ask. It's not at all. And so one of the things that we were struggling with, and I think, Judd, you were the first person to say patients often vote with their feet, is even though on paper and from a regulatory standpoint, this shouldn't have represented a wholesale shift in standard of care, there were a number of reasons why the uptake of this was very rapid, to the point where we couldn't enroll anyone in the US. Fortunately, we were still in the phase 2 enrollment, and so we had some time to pivot, and so we re-engaged with FDA about a new plan, which actually looked at, that incorporated the new, what we now were understanding as the new standard of care in the US. And so we ended up going with an investigator's choice of adagrasib or sotorasib. But as you can imagine, we had some concerns about that because these drugs were still in accelerated approval. And having come from the 2021 accelerated approval ODACs, we were a little cautious about moving forward with anything as standard of care. So we had to work very closely with FDA to really understand if this was possible to leverage these. But we were lucky because we were, we're fortunate enough that we had not gotten far enough along in our development program that we could make this pivot and we could make the change without slowing down the entire development program. And I think about that in terms of our broader discussions on the panel and some of the situations that we brought up on the panel around, well, yeah, that's great if you can figure that out at the very beginning of the trial. What would have happened if we would have enrolled half the trial and then completely not been able to enroll any more patients? And so you have to think about the scenarios where you don't find out about that in time. So it was a really good example of, a lot of times the treatment landscape will inform the standard of care, and it's very clear because you just can't enroll the trials.
I remember those conversations with you, Kathy, and I was really excited to see the trial design that included 2 accelerated approvals. There actually was precedent for this in small cell in our division for lurbinectedin. And I thought it was very patient-centric, and so that was a great collaboration, so thank you for sharing that. And how is that trial enrolling? How did that?
It did great. It's doing fine. And so, yeah, it was
Wonderful.
the pivot was really helpful. And one other thing I would note, because we are talking about global trials and the challenges around multi-regional trials, the other fortunate aspect of that example is that the approvals were fairly, we had broad approvals globally, so we could include that and not have to think about including chemo options in the arm. So we had enough approvals globally that it didn't affect the global approach to the study.
You said you were lucky, the timing worked out, you were able to pivot, but what if you had been further along operationally? What if you had been 25, 30% enrolled? Sumitra, I'm gonna ask you to comment on this. What would Kathy have done? Or what is one to do if they're asked to kind of invoke multiple potential control arms that may or may not be equivalent, if you will?
I think the biggest issue with multiple control arms is heterogeneity of the treatment effect, right? So trying to develop one global trial and having multiple standard of care arms that may not be equivalent in outcomes or tolerability or schedule or what have you makes it very complicated. So what are my options? You could do region-specific trials, and I know nobody likes that because that does complicate how you do the trials. It makes it larger, and at the end of the day, when you have to combine data across those trials to still come up with one overall answer, that makes it very difficult. So I think if you can plan up front that you think you're going to have to pivot, what are your options? Planning up front, I think, is the biggest thing, rather than being in the moment and trying to redesign a study.
And we've heard that that's incredibly challenging. Even Kathy, with all the power and insight that companies have, it's hard to know the impact that will they be approved? What impact will they have? They're under accelerated approval. We haven't yet confirmed benefit, yet patients are voting with their feet. So I'm gonna go to Ray and Kristen on this. Ray, when you see a trial readout that perhaps doesn't utilize the control arm that fits what you're using, first line, second line, and you're a thoracic oncologist, so we'll stick with the theme, how do you interpret those trial results?
Yeah, that's a great question, and it's an increasingly germane question because of the fundamental excitement we all have to remind ourselves of. It's a good thing, right? Rapid cycle development, discovery. We have so many more options. With that has come this complexity. Okay, let's embrace it, right? So as clinicians, we're constantly, of course, remembering who's number one, the patient. And we're trying to figure out, am I comfortable with the options that I have? Am I at equipoise? And in my conversations with my patients, are they too at sufficient equipoise to take the step that we want them to take? And we're constantly having to interpret, right? Because there is no such thing as a perfect trial. In a world of widening options, we have to make decisions in designing and choosing when we do our clinical trials. We get that. We're constantly reminding our junior colleagues when we teach them that we're not supposed to do cross-trial comparisons because of statistical issues there of internal versus external validity, but we do them all the time. Of course, that's the reality of it because you can't ask and answer every question in every clinical trial. So constantly what we're grappling with is, well, how deviant from my world is this control, is this trial? Is this at the extreme of inapplicability and therefore worthy of dismissal? Or can I envisage a scenario within which, yeah, I do see these kinds of patients. Yeah, there are situations in which we have to embrace this option, and therefore there is meaning to me in interpreting the results. So we do have to sort of cogitate a little bit. in the absence of hard evidence. It's the internal versus external validity push and pull.
And we love cross-trial comparisons. I think where would we be without them? So in all their flaws, I mean, what I'm hearing you say is you have to take all the data and kind of you put it through your filter as a thoracic oncologist and apply it to the patient in front of you. Yep. So Kristen, we talked about this in our, prepping. Mm-hmm. What does a patient, what's your perspective on a patient's understanding of standard of care? Whose responsibility is it to inform the patient of what the standard of care is?
Yeah, and in our conversations, I mean, it was very apparent to me that I don't think most patients, I know I certainly would have any idea how complex this issue is, that they wouldn't have any understanding that standard of care could vary by country and even regions within the country. And so, when they're having these conversations with clinicians, the physicians, really need to be transparent about the evolving options and, and helping them to understand that there could be, varying changes that could happen, but to help them inform their decision about whether to enroll in a trial or not.
Jed, let's come back to you about, okay, you're designing a trial, you now have to choose a control arm. We've done all our research, we've decided. What are the major drivers that go into that decision-making up front?
Yeah. So as we think about those control arms, there's a lot that goes into it. I reflect on, on Ray's comment that there is no perfect trial. And so I think first and foremost, we have to accept that, as we're doing this, we're going to have some trade-offs. We try to learn from the past. So as we designed our prostate cancer trials in the phase 3, we spoke to a lot of the clinicians who had participated in those trials and found out what didn't work well and also what did work well. Things that we learned were, in certain instances, based on the control arm, there was a high level of dropout from patients. We know that that will pose significant challenges to the interpretability of the overall results. And so we're thinking heavily, how do we kind of mitigate against that potential dropout? One of the options for that is to add in more options for the patients, give them something that they would want to go on that control arm if they enrolled into that trial, hopefully, kind of preventing that. So, to Sumitra's point, then, the challenge is you're adding heterogeneity into your trial. And so that's one of the trade-offs that we have to think about as we think about an investigator's choice for that control arm. Ultimately, that's what we went forward with for our 2 phase 3 trials. I think the second is looking at, from a company standpoint, where you are trying to seek your approvals in the future. And so you need to make sure, not only, for the United States, as we've talked a lot about today, that the results are applicable to that population, but we're thinking about the other regions of the world and what control arms are utilized there, and will these results be interpreted there. The other piece of it is, sometimes for certain reasons, a drug won't be approved in a certain region of the world, and you want to have it as part of your control arm because it is approved in other places. And so that's another component of it that we have to think about. The final one that I'll just touch on, and I think we have had a lot of discussion about this as a group, is the feasibility of the control arm. There's a number of agents that are out there now that have gotten approval and are considered standard of care: radioligand therapies, CAR-T, other cell therapies, gene therapies, that can be very difficult for a sponsor to deliver as part of the control arm for a trial. And so we also have to look at the practical standpoint of, can we actually even include this and be able to supply it as part of our trial?
Kathy, let's follow up on the feasibility part. How much due diligence do you do ahead of time around feasibility of the control arm and the various potential choices in a control arm? And how do we best communicate where we may be encountering challenges with feasibility to regulators?
It's a very timely question because we just had this discussion with regulators recently. So I think Jed, you did a nice job outlining. So, I would say that a lot of it comes down to talking to the physicians and the key opinion leaders to really understand the treatment landscape where we're going. So Jed, you've really hit on those are the 2 major things that we look at. And certainly, recently, that this was an argument we brought back to the regulators because we ended up with a control arm, an investigator's control arm, that got questioned because the health authority said, we're not comfortable with these two in here because we don't think these are comparable and there's no direct studies comparing these, and this is going to complicate your trial. This is going to be very hard to interpret. But our feedback to the regulators was, well, we're trying to design these trials to reflect a more real world treatment setting. And to your point, Judd, because this goes to the question of feasibility. We want to be able to enroll these trials. We need to have control arms that reflect how patients are getting treated in a real world setting now. And so that's not always convenient when we start looking at what ends up in the control arm as investigator's choice. And inconveniently, that sometimes is going to cause us some downstream pain. And in terms of how we talk about that with the regulators, we had done a lot of mitigation, and so we talked through, here's how we're planning. We're going to cap, we're going to do this, we're going to do that. So we did have that very candid conversation, but at the end of the day, the regulators can only warn us to say, you're designing something that is potentially going to be messy on the back end. But our response was, well, this is just the complicated scenario we're in right now. If we're going to be providing the most contemporary treatments in different regions, we're going to end up with kind of some mismatches in terms of our control arm and our investigator's choice. So those are difficult conversations, and I don't think that there's an easy answer to how we sort through that with the regulators. I don't think that can be answered by just asking it in the right way.
No, and I think the data-driven approach is critical. And one piece of data that's so critical that we talked about that I love that came out of we say patients vote with their feet, but what we came actually out of that was patterns of enrollment is a surrogate for applicability. Ray, do you want to expand upon that? As you're enrolling patients in a trial, regulatory bodies may want a radioconjugate. They may want a CAR T as the control arm or part of the control arm. But you're in the clinic, you're enrolling patients. What do you think about that? Patterns of enrollment are a surrogate for applicability.
Yeah. So the first thing I'll say is, you know what the worst clinical trial is? The very worst clinical trial, bar none, is one that never completes. That's the worst. You wasted every resource there is, including human resource. So the other thing I would point out is that even when a clinical trial is successful, if it doesn't yield results that are implementable, then we have also lost in a way. So I think the idea that we design our clinical trials and we then have to execute them within our clinics is very, very important, and something that we maybe need to give even more attention to than we currently do. Because the barriers are, first of all, if somebody came to me with a clinical trial with an arm that I'm not at equipoise at, as a member of the people that choose what clinical trials we accept or not, that's the first barrier. Chances are we will winnow out that trial. And then the second, of course, is if we go ahead and somehow convince ourselves that this is okay to do, there is the challenge. You do have to sell it to your patients. And, people trust us, of course, but that trust has to be earned. And they don't trust us blindly. You want to do what? Me to do what? For what? Why exactly? And then we get into the world in which we're saying, well, we open this trial, we're not accruing to it. And therefore, we're very sensitive to what clinical trials do we agree. Nobody wants to expend all the resources to open a trial and then see it not accrue.
Well, that goes back to a little bit about the feasibility, right? Trials don't just open at your center. Someone comes and asks you, would you be willing to enroll patients in this study? I want to react to something, and you have the option to say, yes, I think I could enroll patients in this study. I think it would be attractive. You use the term sell, which I think is interesting. Kristen, I want to come to you on this and the issue that Ray's bringing up of trust and how patients choose to enroll on trials. Can you talk a little bit about that decision-making and the trust with the physician and all you've heard about with the standard of care and the control arm? How does that trust extend to the fact that I am being randomized if it's a randomized trial? which it has to be for a control arm, to be to a control arm that makes sense, that there is equipoise.
Right, and I completely agree with Ray's points, and I think, from a patient perspective, a lot of times how they're perceiving how a trial might be for them could mirror what they're hearing from their physician. So, if they're perceiving the physician as thinking one thing or the other, they might lean in that direction. But there could be many factors that are coming into play as well. From a patient perspective, it could be a generational, how they view the physician. It could be cultural background, just to give a couple of examples. And so, the perspective of the physician, I think, is really important in the decision making for the patient about whether they think this could be a feasible option or a good option, or is it going to cause them too much stress, and whether it would be helpful. And again, especially keeping in mind, I think most patients probably don't understand the benefits of research, and not just that it could benefit them, but a whole community. And but as you're facing a health issue, weighing all of those options at one time just brings on a lot of complications.
So, we can't predict the future. I think Ray said this is a good problem, actually, that we have, right? We're having more assets approved, standards of care are changing, but we still have to move forward with our trials. We can't kind of wait and see. We have no magic 8-ball to predict. So if you're entering into a trial, Smitha, this is going to come to you, and you have a maybe 50-50 chance that in the midst of this trial, XXX trials are going to read out, and depending on the magnitude of results and how the regulatory bodies see this, this may make our current trial impractical, infeasible. The patterns of enrollment will in fact shift ex-US more than likely, and maybe more depending on the global regulatory positioning. How could we potentially plan for such contingencies?
I think I will say something similar to what Ray said. Not anticipating change is the worst thing you can do when you embark on a clinical trial today.
Interesting.
Right? I think you have to anticipate change. So having said that, what are some things you can plan for? I mean, as many things as you can plan for, right? You already probably know what those other trials are, comparing against, how it could potentially change your ongoing trial. Maybe it doesn't always have to be that the readout and then you have a new control arm. It could be also that you have to restrict enrollment because those readouts are telling you that you can't enroll all the patients that you have always been enrolling on your trial. So even planning for how do you restrict enrollment. Can you prospectively plan for embedded cohorts in your ongoing study if you know that there is going to be a new standard of care or something that's going to read out that makes it impractical for you to continue your current trial without, an embedded cohort? I know this is nobody's favorite, but you can plan also dual endpoints. It doesn't necessarily fit into this, but also sometimes the readout on those other trials could be on a PFS. If you have a dual endpoint, you could elevate OS to be your primary endpoint. You can still continue going on. You can do stratification on the timing of when these changes are going to occur, so you can look at the patterns before and post the new standard of care that comes out. You can look at region-specific analyses. So there's just a whole plethora of things that you can consider, but I think as much information as you can put. I think in an earlier session it was told also that you have to plan, map out your different scenarios and see where that'll put you and how far along you may be in your trial by the time you have to make these design changes. But planning for it and getting it into your protocol, I think, are the two important things.
Okay, Jed, go respond to
that.
this.
Yeah, I was going to say, I'm going to build on that a little bit from the sponsor side of things. I think this is why oftentimes there's so much, not pressure, but we're trying to enroll these trials as quickly as possible because we look at these competitive landscapes and it is such an exciting time to look at the landscape. But the reality is that something is likely to get approved the longer that we kind of have that trial enrolling. And to Ray's earlier point, the last thing we ever want is a trial where we don't complete it and don't answer the scientific question. That's not a good use of the patient resources, the resources of the trial infrastructure. And so I think that's where kind of all of the sponsors are now trying to open more sites globally. That gets back to needing more countries, having the control arm be relevant more places so that we can complete that enrollment in a timely fashion so that we hopefully get to avoid this really challenging situation.
Kathy, what if we're in a situation where the control arm shifts in certain regions, but not in others mid-trial? I noticed, Sumitra, in all of the contingency plans, I never heard you say wholesale change the control arm, which I like. Maybe you did say it and I missed it, but you talked about embedded cohorts, co-primers,
I didn't
many
say that
options.
because you still have to figure out how you're going to use all the data on the prior control arm. Are you going to restart?
Right.
That's.
So what if it's just impractical? I entered one of our sessions, our working group sessions on the panel. I said, I'd like to propose a percentage enrollment where it could be actually appropriate to change. And I was met with just visceral reaction. And I walked it right on back. So we are not proposing such. But Kathy, it's impractical to change the control arm. Why? And A, it's a two-part question. And B, if you can justify that, how can you provide, what are some ways to provide supplemental data or additional data which may assure regulators and others that the data you've generated is relevant?
So I'm going to build on, Jed, what you said in terms of really in practice how we do this, because we are looking at the entire treatment landscape, and you can see all of us have, we're looking at development planning and seeing everything that could potentially read out. So we're rarely surprised. And to your point, the most likely region where this is going to occur is the US. It's got the accelerated pathways, and so we're tracking those carefully. And I know, Jed, if you're like us, we have those starred everywhere where those are going to show up as potential approvals. I think that, fundamentally it goes back to, it would be fundamentally, if you're starting your trial, that's a key consideration, whether or not something is going to read out near term or halfway through your trial, because, and it's always going to depend on what that emerging data looks like, how confident we are that that would actually have any kind of impact on the overall. And we talked about this on our working group, that not everything is going to result in a shift in standard of care. A lot of things are incremental benefits, and they just add treatment options, and your standard of care may still be very relevant. So that's one of the fundamental questions we ask: is there something that's emerging that could shift? But to your point, what do you do? And I think you're asking, so we didn't see this coming. And I think this was in some ways was the KRAS if we just happened to catch it early, but this would have been the KRAS example if we would have gotten 6 months into enrollment. And then the question would be, do we pivot and redesign that trial? And quite honestly, that does keep us up a little bit at night to think about, did we read something wrong in that? But I think, Jed, to your point, we do see this, and there is some expectation when we have conversations with the regulators, and probably even more so now, we're going to have very direct questions with the regulators to say, do you agree this is what we're seeing in the treatment landscape? Do you agree this represents the standard of care right now? We can't predict how those trial readouts, and we don't know if we're going to get a Pembro reading out halfway through and suddenly be in a sticky situation. But for the most part, we have to engage with what we know at the moment and what we can reasonably predict is going to happen during the course of the trial. And so I think that's what we have to move forward with.
Yeah, and I think one nuance that may not have been introduced is the idea that it may not be one transformative approval, right? There may be a situation in which there's a class of drugs. I'm thinking second-line X malignancy, where there have been 2 or 3 checkpoint inhibitors approved, each kind of eking out 6 weeks, couple months survival benefit, where the FDA may say, this class of drugs, patients should be getting this. But there could also be a situation where investigators are still willing to enroll, and patients are still willing to enroll on the trial as designed. So I think this gets into a very interesting dance between patient centricity, regulatory expectations, feasibility, completing the trial. we're having some very interesting questions come in. I think, Judd and Kathy, I want to stick with you just a little bit longer. Why is this such a critical topic, and why is it such a critical topic now? What are the implications, potentially?
Yeah, I mean, I think as somebody leading drug development, the biggest challenge is the interpretability of your results at the end of that trial. If people don't believe that you use the right control arm, they may do the cross-trial comparisons to try and figure out whether or not this is a viable product for their patients. But at the end of the day, if we haven't answered the question for them and haven't given them the appropriate data set, then we haven't really done our job as drug developers. We haven't advanced it in any way, shape, or form in bringing that medicine to patients. So, I won't say that's what keeps me up at night, but that is, the anxiety, right? Is this the right trial? And I think that's why we spend so much time talking to clinicians and patients, around the globe to make sure that if this is a positive trial, that this would be a meaningful kind of change for them.
I think that's a really nice perspective to share, because certainly there is the perception, which we talked about in the working group, that we just go for the low-hanging fruit, for the least effective standard of care just to get a win. Well, that actually would make the trial likely impractical, infeasible, and physicians like Ray in the clinic would say no thank you to this trial at his center. Fair? So Kathy, anything to add to what Jed is saying, and then I'm going to come to Kristen on this.
Yeah, I would say, just to build on what Jed said, is we learned a lot. We had these transformative therapies come through with immunotherapies, right? So we've learned a lot, and I'm reflecting a little bit on the last panel discussion and on the comment by Amy and Roy about they're anticipating the next advancement, the next acceleration, and so I think really why this is so important now is we want to have this problem. We want to have this problem forever. And so we need to figure out how to manage this problem because we want to keep having this problem. We need to figure out how to design trials. And then going back to the overall theme of that last discussion is we need to be able to design trials that we can read out that are patient-centric. There's a lot of things we're trying to do. So I think what's really important and why this is so important now is maybe there's a little bit of a lull before we see another class of drugs come through where it's transformative. And we do need to make a really fast pivot right now. We're doing a lot of add-on therapies, and maybe this is incremental gains that we're seeing right now. But we need to be ready to be able to not disrupt drug development when we see the next advancements come through. So I think it's important to really address it now.
Kristen and Ray, how can industry do a better job of incorporating patient and provider perspectives in protocol design and development?
And I think it can vary by patient. I think some patients might be excited to hop right on the newest therapy available, whereas other patients might prefer some more stability as they're making their decision. It's been well established today how much and how quickly the science is changing, which is wonderful, though I can also imagine for clinicians and researchers and everybody, the influx of data that you're receiving, how challenging that could be to digest and then share with patients. And someone on an earlier panel mentioned how important it is to have the informed consent be clear and understandable for patients when they're making these big decisions about whether to enroll in a trial. And then, the line of communication between the physician and the patient needs to be ongoing throughout the process if the standard of care is changing. And, that might be revisiting that conversation about how the informed consent could be changing.
Right.
Yeah, I think my response is very, very simple. Out of the New Testament, seek and ye shall receive. Ask and you shall. Is it ask and you shall find, seek and you shall receive?
I have to brush up on my New Testament.
You just need to ask. Ask early, when there is still the opportunity to make a change. So I'll take the example within SWOG, which is one of the National Clinical Trials Research Bases. In our long committee years ago, we acknowledged that we needed direct input. So we did two things. One was we decided no protocol will move forward without the active engagement and feedback from a patient advocate. And the second was no protocol shall move forward without the direct feedback from the community providers who do not necessarily come to the meeting or sit around the table when we're planning these things. And that has made a big difference. And I think in choosing control arms, in trying to figure out feasibility and things like that, what all of us need to do is ask.
Including regulatory bodies. Ask. So we're coming down to time. We have a ton of questions, very interesting questions. So rapid fire responses. What's one practical next step to make comparator planning in global trials more predictable and collaborative? Jet.
Wow, tough one to go first on for the rapid fire. I think it comes back to kind of being patient and customer centric, right? At the end of the day, if you pick your control arm because you believe patients will enroll on it and want to be on that just as much as the investigational therapy, that to me is the number one.
Planning, planning, planning. I think you have
Call
to
your
plan.
statistician. Kristen.
I think, it's communication. Communication about evolving standards of care between the physicians and the patients so that in partnership, and I really think that's a key point, that they're making these decisions and everybody has the full understanding.
Right.
Simplify, simplify, simplify. For example, make it easier for the patients and the clinicians who have to guide their care, trust them. Therefore, things like dealer's choice, yes. Embrace the full range of standards that people practice in and make your trials more externally valid.
And we see plenty of examples of dealer's choice, and I think the complexity certainly comes when those choices are quite different in their efficacy or certainly mode of delivery. Kathy, one thing.
I would say more multi-stakeholder discussions and focus on this because I think bringing the regulators, the patient, the treating physicians and sponsors together to really understand the most difficult scenarios we're going to encounter.
And I will take the liberty of adding, I will say information sharing with the regulators. I was moved as a former regulator by data that was brought to me on patterns of practice, what was happening in the US, and in light of approvals, what was actually happening in clinics, in community settings, and I do think that that's really critical data to gather. Okay, so let's turn to audience questions. This actually has come up. And I'm going to flip it around, but the question is, well, when there are multiple standards of care, can you clarify if the FDA requires a therapy to be compared to the best available standard of care or just to one of many standards of care? Should I answer that?
We should.
You do that.
I think, so again, this goes back to the many standards of care and whether or not there's one that's transformative, most utilized over others, and again, I think data can support this, but usually it's about a class effect, but certainly in the case of the RAS drugs, we saw that patients were voting with their feet over docetaxel. There was a question about companies paying for, I think it was question 3 or slide 3 that I thought, maybe go up one, but the question was about, and this does come up, if a comparator arm is not approved in other regions, why can't the companies just pay for that? This is it. When a therapy is not yet approved or covered in other countries, why don't companies just provide it for the patients in the study?
I can take this. We had this come up recently in prostate cancer. So there's often a reason as to why it wasn't approved in that country. So it may be that it hasn't been demonstrated to be efficacious and safe in that patient population. So us just providing something that's not considered a standard of care in that country you could enroll there, but that doesn't mean that that health authority would then perceive that as a standard when doing the comparisons. But it's a good question.
Okay. All right. Seamless designs seem to introduce an interesting tension into development as you are choosing your registrational trial comparator years before the planned submission. Do you have any recommendations on how companies can ensure their original selection of a comparator is still relevant years down the road? It's quite a challenge, I think.
But is it seamless designs? I think in the morning the panel really talked about how it can evolve, right? So I do think since you have picked a comparator, for your phase 3, right when you're doing your phase 1 or your phase 2, I think there's still an opportunity to pivot there. I think that it's harder when you design your phase 3 and your comparator arm actually changes midstream, which is what we were discussing. This one's like you're planning up front. I think you can plan for this.
How can label and regulatory philosophy be optimally adjusted to allow more flexibility in comparator arms? I think there's many oncology approvals with investigators' choice control arms. I don't think that's, again, I think the real nuance is when those choices vary substantially, and we've encountered that. And that's what makes it just incredibly statistically complex. Amy McKee in the last panel said, I'm just going to tell you guys, it's one trial. We're going to do one global trial. We have you here. Do you agree with this assertion? And if so, why yes or no? One global phase 3 trial. Why can't you do 2? Why can't you do, it's been proposed, it's in the multi-regional guidance for what it's worth, just do 2 trials. Just do one in the US against the US standard of care and do one ex-US. Tell me why that can't happen.
Are you directing me?
Industry.
Yeah, I will address this because we've actually worked up some CDPs that have considered, especially pairing maybe a pragmatic design in the US, looking at a more updated standard of care with a global trial, and we've discussed these with OCE in the past. So these are certainly feasible. I think when it comes down to looking at the statistical, how we pool the data, I think these become a bit complex and they're not as attractive sometimes internally because we've got more complex designs, and I think thinking about how we handle that data on the back end, it's the same as the investigator choice control arm where you've got 2 different, so you have the same downstream implications of data that may be somewhat hard to interpret. But we have played around with the idea of doing that.
You've played around with it.
We played around with that.
Okay. Sounds like that's where it ended.
It's landed there.
I guess just to add to it, it comes back to the reason of why we do multi-regional trials, right? The fundamental reason is so that we can try and bring these medicines to patients more quickly, and establishing kind of concrete evidence for their use. So if we go back to doing a US-only trial or back to only doing a European or, in Asia, the challenge then becomes every one of those is going to be slower, and then the medicines,
Can we delay access?
Exactly.
So effectively what I'm hearing you all say is that the most rapid way to provide access to novel therapies is one harmonized phase 3 global registrational trial. Enroll it as quickly as you can.
Yeah. Time is money, time is life in what we're talking about. And what we also increasingly are faced with is the population is no longer monolithic like it once was. It's smaller and smaller fragments of the block. And we are now tasked with finding those people where they are, not where we choose to look for them. And as we keep pushing in that direction, the force on us is going to be even greater in that direction, I think.
I'm going to take moderator liberty and end it there, Ray. Thank you so much, and thank you everyone for your time.
Thank you very much. You're more than welcome to exit the stage, I suppose. I don't want to keep you here. No, thank you all very much for a thoughtful discussion on a complicated topic for sure, and thank you, Dr. Singh, for moderating. I'll wrap up today's meeting very quickly and just say simply thank you. Thanks to all of our panelists, to all of our moderators for such engaging conversations today. Thank you for everyone for joining us in the room throughout the day and for being online for those of you who joined virtually. This working meeting has really been a critical forum for advancing our work as an organization as well as a community more broadly. In order to collectively catalyze the future of cancer research. So thank you for your contributions today and leading up to the meeting. I want to thank everyone that helped make this meeting possible, certainly to the amazing group of people at Friends of Cancer Research who made all aspects of the meeting today happen. A special thanks to our speakers and working group members who provided their time and expertise to help address the complicated topics that we were able to raise today and hopefully move forward through these discussions. Moving forward, we are looking forward to a very action-packed 2026 with multiple events and data readouts scheduled. This will start with a forum that we'll be hosting on February 5th next year, focused on modernizing oncology endpoints. The meeting itself will include new data from the CT Monitor project, exploring the use of ctDNA as an intermediate endpoint in drug development. We will also be discussing plans and initial findings from Friends' AI Resist project, which is advancing the use of automated imaging tools and design of new endpoints to increase the efficiency for future clinical trials. You can find more information or register for these events on our website under the Events tab, and we hope that we will see you again in the new year. Thank you again for joining us today. Safe travels, and good to see everyone.